there's no dash.
Okay. There's no dash?
There's no dash. It
Okay.
just is long, so.
Oh, I see.
Yeah.
Thanks.
Good.
I think we should be good. Okay.
All right, we are ready to start. Welcome to the Research Fest of Parallel Square Technology Institute. PTI is a non-profit research organization aiming to accelerate biomedical research through proteomics. We think that direct measurements of proteins at high scale, high resolution, and high sensitivity hold the key to making fundamental discoveries in biology, to characterizing pathologies, and ultimately contributing to developing more effective therapies. So let me share with you our perspective, why we think proteomics is so potential and has transformative capabilities. About 200 years ago, many people believed in a vital force called elan vital. And this force animated all living things. And then careful experiments demonstrated that instead of elan vital, there were proteins that were performing all of the functions that we associate with life, such as fermentation, biochemical catalysis, and we could isolate, purify, and even crystallize these proteins. And the ability to isolate and purify proteins drove biochemical research for many decades of the past century. This was the mainstay that established many of the fundamental conceptual frameworks that we have in biology. Are biological macromolecules amorphous, or are they molecularly defined? Are protein conformations and structures imprinted by some templates, or are they determined by the primary amino acid sequence? And not only fundamental research, this ability to characterize isolated types of proteins also contributed to improving human lives. The determination of the primary amino acid sequence of insulin ultimately resulted in improved standard of care for millions of people suffering from diabetes. And then in 1980s, a new technology that allowed efficient ionization of large molecules, converting large biomolecules into ions, provided a very significant improvement in the scale at which we could analyze proteins. So that instead of analyzing isolated single type of protein, we could analyze more proteins. In the very memorable words of John Feng, it conferred electrospray wings to molecular elephants. And this ability further increased thanks to more technologies empowering the analysis of these ions by mass spectrometry, such as developments of detectors, the Orbitrap, development of time-of-flight detectors, recently Astro, and technologies that allow to separate and accumulate ions, such as trapped ion mobility spectrometry. All of these technologies gradually increased our ability to analyze proteins at larger scale. And very important to this development were organized efforts by the scientific community to introduce rigorous benchmarks and community standards as part of initiatives such as Sipe that ultimately resulted in generating large-scale data sets suitable for clinical research and improving our understanding of diseases. In the meantime, of course, there's another very important set of technologies that have been contributing to biological research, and this is the ability to analyze nucleic acids, particularly to sequence DNA from the first generation of Sanger sequencing to the second generation to the third generation ion pores. This has been an important contribution driving biomedical research over the last couple of decades, over the last few decades. And because these methods have become so accessible, they're oftentimes used to provide indirect information about proteins as well. And we can easily understand to some extent why those technologies have scaled more readily if we consider the relative complexity of the genome, the transcriptome, and the proteome. In a typical mammalian cell, such as a human fibroblast, there are mere thousands, less than 1 million messenger RNAs. There are billions of protein molecules, billion with a B. So there are many orders of magnitude difference in the complexity, in the dynamic range of proteins, of their modifications, of their interactions. They're so complex that we're yet to identify and characterize all of them. We don't know all of the proteins making up our bodies. We've never been able to identify them. We've never been able to characterize their primary amino acid sequences, not their modifications, not their interactions, not their functions, not their localizations. It's a tremendously, tremendously complex system that is very challenging. It poses a major challenge, but I also think it's a wonderful opportunity for scientific discovery and for improvements of medicine. So, let me tell you why I think measuring proteins directly is important. There are many reasons. I'll share with you a couple. One of those is if we're interested in describing just protein concentrations, protein abundance, what are the regulatory mechanisms that determine the concentrations of proteins, different proteins in your cells, their differences across different cell types and tissues? We find that about half of that is determined from the synthesis side of the equation, and the fully other half is determined by regulation of protein degradation. And if we want to be able to measure and characterize protein degradation, we need to directly analyze proteins. It is very difficult to make these inferences from measuring nucleic acids alone. But even when it comes to the synthesis part of the equation, it turns out that if we want to know what are the primary amino acid sequences of proteins, there are in vivo mechanisms of alternate translation guiding which amino acid is incorporated to which codon that still require direct characterization of proteins, direct measurements of proteins to know what is the proteome that is made in human cells in vivo, as opposed to predicted indirectly from models. And of course, a very, very important reason for direct analysis of proteins is understanding human diseases. We know that for many diseases, the central contributors, the central players, are protein pathologies. Whether these are modified tau proteins and aggregates in neurodegenerative diseases, whether these are aberrant protein kinases contributing to cancer, proteins are central players in diseases, and we need to have the capability to specifically first characterize what are these protein modifications, protein aberrations, and changes associated with the disease. And because human communities are large and complex and heterogeneous, we need to have measurements at sufficiently large scale that can deal with the associated comorbidities and variations across the population. We need measurements that are specific, and this might seem like a simple thing. It has been a very difficult thing to achieve in the world of proteomics, specific measurements. Many of the current technologies struggle still to achieve specificity. We want to know what we are measuring. And then we need technologies that are affordable, that we can really do this for many diseases for the population. Beyond simply identifying associations, we want to transcend the descriptive nature of those and just finding biomarkers. We also want to understand the mechanisms contributing to disease, because the closer we understand the mechanism, the better chance we stand at ultimately developing effective therapeutics. So all these reasons motivate us to think of how we can overcome the substantial challenges of the proteome. And they motivated starting PTI about 2 years ago. So we're a very young organization. We have grown from just a couple of people to about 20 now. And we are very excited today to share with you some of our initial results from developing technologies to enable proteomic analysis, applying those to biological problems that we think are important and impactful, trying to understand mechanisms contributing to disease. But I think as we share with you today our progress, it is worth keeping in mind that everything that we share is work in progress. We have been working on this for 2 years. For some of the projects that we'll share today, we've been working for mere months. But we are nonetheless very excited to share the progress that we've been able to make, because we think it is beneficial not just for us, it is something that we would like to empower others to use in their research. So how do we approach the formidable problem of scaling up proteomics, of analyzing proteins? We appreciate and embrace all technologies that can help us with this problem. I think there is scope for a lot of different approaches to contribute. But in particular, we think that mass spectrometry is very powerful, and we have a defined strategy of how we can use it. For a long time, mass spectrometry has been well established as being very specific, very versatile, very powerful, but also has some major weaknesses that has prevented it from contributing significantly to biomedical research. Namely, it is relatively difficult to scale. It is relatively expensive, and we want to address those issues. We want to have sensitive methods that can be scaled by orders of magnitude, that can have sufficient sensitivity and be sufficiently affordable so that they can really make an impact in the real world, not just in research and development, but also in helping develop better cures for diseases. So, how are we going to do this? Conceptually, the approach is relatively simple. We want to be able to parallelize data acquisition. The more data points we can acquire per unit time, the higher the throughput, the more scalable the approach. And the way we are doing this is by parallelizing simultaneously in multiple dimensions. One dimension for parallelization is when we analyze peptides or proteins, instead of analyzing one peptide at a time, we can isolate and fragment large number of peptides. Another dimension which we can parallelize is by being able to barcode molecules from different samples, then mix them together and analyze the barcoded molecules at the same time. And if we're able to combine those two axes of parallelization, then we get the product of those axes. We are able to increase the number of data points that we can acquire simultaneously. And this roughly provides a framework that I can use to illustrate at this very high conceptual level, welcome David, some of the technologies that we'll share with you today. So one important thing that you can quickly realize is barcoding. We need to have molecules that allow us to barcode multiple samples so that when we mix them, we can do this analysis in ways that don't compromise the quality of the data. And in fact, today we are going to share with you two exciting advances, exciting for us on that front. One is making new molecules for doing this barcoding, and the other one is a conceptually new approach to multiplexing samples in a new dimension, in the dimension of time, as Derks is going to share with you. And as you can imagine, acquiring more data simultaneously sounds good. It's not a piece of cake. It also has challenges. So we have much higher density of data points, and we also built in more structure in the data to really solve the challenge of data density and take advantage of the structure in the data that we build. We need new algorithms. And we've made developments in that direction that McDonnell is going to show. So as we scale the data, increase throughput, decrease cost, we are very, very keen to increase confidence and quality in the data sets. This is not just about let's get more data, we can deal with poor quality. No, we want it all. we really would like to have very high quality in sequence determination. As I already mentioned, simply determining amino acid sequence rather than inferring that from nucleic acids is a challenge. So we want to have confidence in determining those sequences, in those modifications. And to just give you a conceptual flavor of one of the important things that we keep an eye on. So if we want to determine the sequence of this peptide, each letter corresponds here to an amino acid, we break it into pieces, and then we can use the pieces to put together the puzzle. And if all of the pieces are present, we can do this with high confidence. But if we have missing pieces or fragments, then it becomes hard to put it together. And you can see that when we analyze this peptide without any label in isolation, we are able to see quite a few fragments and have a good score. But when we label that same peptide with a barcode that we made at PTI with the help of our collaborators, we are able to identify more fragments, that is more pieces of the puzzle, so we can determine the sequence with higher confidence. We can better localize post-translational modifications such as phosphorylation, and we can better do de novo sequencing, which I think is very important for us in a number of contexts, for identifying new antigens, for discovering new forms of alternate RNA coding. So we are very excited to be thinking of not only how do we get more data at a lower cost, but how do we improve the quality of the data that we generate. And one thing that I think will be apparent today from our presentations is the deep synergism between the things that we've done. We develop new ways to parallelize, that creates new data structures, new challenges. We also need to have new software to decode that data, to take advantage of that structure, to solve the new challenges that we created when we increase data density. We also need to tailor the biological applications and questions. We need to design our data acquisitions according to the biological questions that we ask. So there's a very deep synergy between these processes. And this is an aspect that is very dear to my heart at PTI, that we're not focusing just on the technology, we're not focusing just on biology. We think that a lot of key fundamental biological questions cannot be answered unless we develop the technology. But which technology, how we develop it, that is informed by the biological questions, by the desire to better understand pathology. And we really put a lot of emphasis on the synergy between these projects to make the whole work better than individual parts would ever work in isolation. And as I stated at the beginning, our primary aim is to advance biomedical research, to accelerate the pace of biomedical research. And we cannot do this alone. Doing it really requires sharing our technology and methods with the broader community. This is a massive, massive ambitious goal. And we realize that we need partners and we need to empower others to use our technology. So we are very passionate to be able to share and support others using the technology that we've developed. I think there are plenty of problems that can benefit from it. And we are very fortunate to be philanthropically funded by visionary supporters who understand the importance of data sharing and support our desire in empowering the community. So we put a lot of effort, not just in tossing a data set here, you can go figure out what it is, but we put effort in describing what we have done, creating metadata that are useful so that the data can be reused, and also organizing information sharing, such as this symposium today, where we aim to broadly enable the community to use what we have created. And of course, I would like to thank all of our supporters, certainly Convergent Research, that have played an important role in facilitating the formation of PTI, and Eric Schmidt Features, and Ken Griffin for providing funding to support our bold foray into making proteomics more powerful in the service of accelerating biomedical research. Now, before I invite Harrison here to introduce the first speaker of our first session, I do have a request to make from you. We had a wonderful group of people here, very diverse group of people, also many people online who are watching this. And some are leading experts in the technology, some are leading experts in the biology, and there are many people who don't have deep expertise in one or the other. And we want to make these presentations as accessible as possible. So to do that, I need your help. I need you to help ask questions about all the jargon that we might slip into using. I try to avoid that, but the truth is it's hard. There will be some jargon. So please keep us in check. Ask questions of what is not clear, because our goal here is not a showcase. Our goal is communication. And to communicate well, we want to make this symposium as interactive as we possibly can. And this requires your participation. So I'll stop here and invite Harrison to come and introduce the first presenters.
Good afternoon, everyone. Super excited to introduce our second two speakers. This presentation will be a tag team, and I think you'll understand the pun when we get to it, between two speakers who are intimately familiar with the details and explorations that we've undertaken to achieve some of the accomplishments that you'll see this afternoon. Very excited to introduce to you Maddy, who comes from a background in chemistry and supercritical liquid chromatography before learning more mass spectrometry than she probably ever thought was possible when she joined PTI a little over a year and a half ago. And then I'm also excited to introduce Mark Adamo, who came to us from Dartmouth College, where he managed not only one but two labs, bioinformatics pipelines, and is now working to achieve similar and greater with peptide identifications at PTI. So please join me in welcoming Mark Adamo and Maddy.
She told me that too. She said she was good after that.
Well, thank you for the great introduction, Harrison, and thank you all for being here today. As Harrison mentioned, today we're presenting on our mass tags. The title of our presentation is approaches to increase the multiplexing of proteomics data acquisition, more specifically creating new mass tags to first run more samples in parallel, a process that we call multiplexing, and also to improve spectral quality, which is the quality of data used to identify and quantify peptides and proteins by mass spec. So we're going to start off today's presentation with a biological example. And the biological example that we've provided is one of neurodegenerative disease, because at PTI we're interested in applying our novel proteomics technologies to Alzheimer's disease. So in this example, we have a human brain, and whether this brain is diseased tissue or whether it's healthy tissue, the brain is composed of hundreds of billions of individual cells, and each of these cells contains 20,000 to 30,000 proteins and may contain hundreds to millions of proteoforms. In biology, we'd like to be able to characterize entire tissue regions, such as the brain, but also be able to zoom in and look at the individual single cells within these regions and understand their proteomes. You can imagine this is a pretty large scale of analysis that we'd like to be able to do. Current approaches are not able to handle this scale. If we were to take a cell or maybe a group of cells from that brain tissue, and we were to run it on an LC-MS system in a core facility, it would cost roughly $150 per run to do this. Now you can imagine if we were to unpick a human brain cell by cell, this would be pretty prohibitively expensive, not to mention extremely time consuming. If we introduce approaches where we can plex samples together, or we can take different samples, we can chemically label or barcode them, combine them and run them together on the LCMS, you can see how the cost per sample and also the time per sample would decrease, allowing us to speed up the pace of biological analysis. At PTI, we're really interested in different technologies that will allow us to increase the throughput of proteomics with mass spectrometry. And when we think about this idea of increasing throughput, we often come back to this guiding principle, which is that throughput is equal to the number of parallel peptides times the number of parallel samples. And this is not the only time today that you'll see this proteomics pyramid to introduce this concept of throughput. So we can think of existing proteomics technologies that tend to increase one term of the throughput equation and not the other. For example, really high in the parallel samples dimension is this example called 35-plex DBA. And in this example, we can take 35 samples, we can label them and run them all together in the mass spec at one time. So we're really able to generate a lot of parallel samples. However, we're only able to analyze one peptide at a time using this method, and that's because we're reliant upon DDA-MS, or data-dependent acquisition, which is a mass spec acquisition scheme that allows us to analyze peptides individually and serially. Now, moving across the pyramid, we can see an opposite approach that allows us to really parallelize peptides. This approach is called label-free DIA. We're only able to run one sample at a time with this approach, but we are able to get a greater depth of proteome coverage because we're using a data acquisition strategy called DIA, or data-independent acquisition. And in this acquisition type, all the peptides or precursors within a particular M/Z range are analyzed at the same time using this method, giving us deeper proteome coverage. At PTI, we'd like to be able to create multiplexing technologies that allows us to parallelize samples and parallelize peptides, achieving these multiplicative gains in throughput. There are a few such examples, or a few examples that are already able to combine parallelizing samples and parallelizing peptides to some extent. And one example of this is 3-plex DIA. You might be familiar with this. This was the technology worked on by Derks and Professor Slavov's lab. And in 3-plex DIA, we're able to run 3 samples simultaneously while maintaining that depth of proteome coverage. Guided by 3-plex DIA and these other approaches, we hope to reach n-plex DIA. In this talk, we'll specifically talk about the mass tags that we're creating to enable this. Now, before we talk about the mass tags we've specifically created for this purpose, I think it's helpful to talk about what are the current state-of-the-art mass tags that enable 3-plex DIA. Well, this tag is called M-track, and you can see here this is its chemical structure. An M-track is a chemical label that gets conjugated to different proteome samples, allowing us to run 3 samples in parallel. M-track comes in a set of 3, so you can see it comes as delta 0, delta 4, and delta 8. And these delta numbers refer to how many heavy atoms are contained in each version of M-track. Now, in a practical application, each of these versions of M-track is labeled to different proteome samples, and then those samples are combined and run all at the same time in the mass spec. And we can tell which peptide belongs to which sample based off of its position in M/Z space. Looking forward to the technology we're interested in creating at PTI, we'd like to go beyond the state of the art to increase the plex size. So this will allow us to hopefully be able to multiplex more samples together, but also increase or maintain the quality of data. Now, I think it's important or helpful here to say, or to define what we mean in this context by quality of data. In this context, we're referring to the quality of data as the quality of peptide fragmentation spectra. And this is a peptide fragmentation spectrum here, and Nikolai also showed one of those in his talk. And so if you're unfamiliar with peptide mass spec, what happens is a peptide is isolated in the mass spectrometer and it's fragmented, forming these resultant peptide fragment ions, and they populate a spectrum such as this. And it's the different identities of these peptide fragment ions that help us figure out what is the sequence or identity of the peptide present in the mass spec at a given time. So based off of the fragments present in the spectrum, we can infer that this is the sequence. And you may notice that there are different colored peaks here, or annotated peaks. The blue ions correspond to B ions, which are peptide fragments that contain the N of the peptide. And as you can see there, there's a little caret. So, for example, VA has a little caret, and that means that that N-terminal peptide fragment is found in this particular graph. Now, there are also red ions, or red peaks here, and these represent Y ions, which are conversely peptide ions that contain, or peptide fragment ions that contain the C-terminus of the peptide. And learning B and Y ions will be helpful for later during Mark's portion of this talk. Now, there are certain elements of a peptide fragmentation spectrum, such as this, that can help us help aid in identifying the peptide sequence. We call these elements of spectral quality. One of these is the number of annotatable peptide fragment ions. The more B or Y ions present, the easier it will be for us to figure out what the sequence of the peptide is. Secondly, what is the relative intensity of these annotatable peptide fragment ions? The more intense each of the peaks is and relative to each other, the easier it will be for us to figure out the peptide present. And lastly, what is the complementarity of annotatable peptide fragment ions? For every bond breakage in a particular at a particular spot in a peptide, is there either a B or Y ion present? And so having these elements can really help us assign what the peptide sequence is. Now, tying this back to our ideas of increasing throughput, we want to either preserve or increase spectral quality when we conjugate our new mass tags to peptides.
Please
because we think
increase
this
the
will help
microphone input
increase
volume
our
and
throughput.
ask the speakers to speak louder.
Or it will be essential to increase our throughput. If you recall here, we have our throughput pyramid, and if we reach the top of, our goal is to reach the top of the throughput pyramid, parallelizing peptides and parallelizing samples. And here I've also pointed out where 3-plex VIA lies on the throughput pyramid. Now, if we increase the number of samples we run in parallel with our mass tag, but we see an accompanying decrease in data quality or spectral quality, we can see that we're actually not increasing throughput. Because while we might be increasing the number of parallel samples, we'd be decreasing the number of parallel peptides. So the throughput equation would not be satisfied. So if our new mass tags reduce spectral quality, we're not increasing throughput. We'd like to be able to maintain our data quality, maintaining the number of parallel peptides, or increase our data quality, increasing the number of parallel peptides as we multiplex more and more samples. And this will help us reach our goal of multiplicative gains in throughput. So to reiterate, but also to orient us towards the research portion of this talk, today Mark and I will be talking about creating new mass tags to first multiplex more samples, and secondly, to improve spectral quality. Now, the first thing to do when we were setting out on this journey of testing out new mass tags was to explore what type of small molecule compounds could serve as a good mass tag. And so we worked with the pentalute group at MIT, and we started off with these 2 model peptide sequences. Now, on these model peptide sequences, a number of mass tag candidates were generated. And you can see here that each of these tags contains 3 components. In green, we have a trifunctional building block, and then on top of that trifunctional building block, there were 2 spots where we could derivatize. So the red represents a carboxylic acid derivatization, and the blue represents a boronic acid derivatization. This allowed us to form a bunch of mass tags combinatorially, which was great because it allowed us to explore a sufficiently large chemical space with a relatively simple experimental design. The next thing to do from here was to test how do these candidate mass tags, how do they impact our ability to sequence these model peptides? So what is their impact on the spectral quality of the given model peptides? We ran these model peptides labeled with these new mass tags in the LCMS, and the results here are in this graph to your left. And you can see on the each of the data points or points on the scatter represent a different mass tag. And on the y-axis, we have the Andromeda score of 1 peptide. And what this is essentially a measure of is how easy was this peptide to sequence when it was conjugated to that mass tag. In other words, what is the spectral quality of this model peptide when it's conjugated to each of the individual mass tags? The higher the spectral quality, the higher the score. And the same goes for the x-axis, but this would be that same spectral quality measure, but for the other model peptide. Now, we were really interested in seeing whether there were any mass tags that could increase the spectral quality of both model peptides. And you can see that there's one highlighted up in the corner in red. And so seeing its ability to improve the spectral quality of both model peptides, we wanted to look at it a little bit further. And so here you can see this is the sequence of that mass tag that is highlighted in red there. And it's called PSM tag at PTI. And here you can see its structure and where it would be conjugated to a peptide. The next thing to do was to understand how can we use this tag for multiplexing? Where are the different sites on this tag where we could install heavy molecules? We did this in collaboration with Dave and team at Creagen. And if you'll recall, using heavy atoms was the mechanism by which we can multiplex using MTRAC. So this was an important thing for us to do with PSM tag as well. And with Dave's and the Creagen's team's help, we were able to produce a 9-plex and a 5-plex set of reagents with this mass tag. The next thing to do after coming up with this doping scheme was to also think about, well, what is the impact of PSM tag on a human proteome? Does PSM tag also increase the spectral quality of a proteome? Because ultimately we are interested in applying PSM tag to the proteome, and so we wanted to make sure that its positive qualities from this model peptide screening could be extrapolated onto the proteome. So we applied PSM tag to the proteome using a standard NHS ester labeling, which is pretty well known for well-known chemistry for labeling proteomes with small molecules and mass tags. And this typically necessitates using a relatively large molar excess of mass tag to peptide. And so what we found when we did this initial reaction was that a lot of the unconjugated tag would collude with select peptides within our peptide gradient due to the relatively larger size of this molecule relative to other mass tags that are available. Fortunately, we were able to alleviate this using ion mobility to effectively separate the excess tag from the peptides. And we did this on both our thermo instruments with FAMES and our timsTOF SEP with TIMS, or trapped ion mobility. And this figure is able to show us that we're able to analyze peptide ions or isolate our peptide ions separately from the tag byproducts, allowing us to analyze all of the peptides that we're interested in without analyzing our tag byproducts. So next I'm going to hand over the microphone to Mark, who's going to talk a little bit more about the impact of PSM tag on the spectral quality of the proteome.
Thank you, Maddy. So following those optimizations that Maddy was just talking about, as well as many others, we've arrived at a point where for a single DDA run, label-free and a DDA run using the PSM tag label, we can achieve roughly equal amounts of peptides identified. So I'm showing unique peptide sequences identified in either sample here. And if we focus in on the intersection here, these are peptides that were identified in both a label-free and a labeled condition. So if you take this intersection, we can begin to evaluate in a more systematic way whether the score performance that we saw with our two original model peptides that we used to select this compound actually translates to a larger set of peptides from the human proteome. So each of the points in this scatter plot is going to correspond to one unique peptide species that was seen both label-free and tagged. And from that we can compute both its ratio of relative abundance on the x-axis, and that shows us that our two samples on the whole are relatively evenly matched by intensity. And we can look at their ratio of score on the y-axis, which shows a distinctive shift in favor of the tagged peptides. And this equates to a median 15% score increase of the tagged peptides versus label-free. So this annotated spectra plot may look familiar if you've been paying attention. And to give a little bit more context to this was not entirely cherry-picked. It was selected from the center of that distribution that I just showed. So in an attempt to illustrate what an average score improvement as a result of the addition of PSM tag looks like on a spectral level. So these two peptides, these two spectra, happen to share a complete Y-ion series. You can see it in red. These are the C-terminal fragments, and they match up fairly well. The B-ion series, going to be the N-terminal fragments, are shifted by the addition of the tag mass on the N-terminus. And we also see additional ion information in the B-ion series introduced as a result of the tag addition. So we see this B1 ion, which is fairly strong, and we also see the higher B ions, B5 and B6. And it's the addition of this new information from the B ion series that is sufficient to increase the score of this peptide spectral match from 34 to 40. And if we are to aggregate all peptide identifications from our human proteome sample, then we can actually observe that the pattern of fragment ion generation that we just observed in that isolated example is actually reflective of a broader pattern of fragment ion production across all the peptides in our data set. So here I'm plotting the relative frequency of fragment production for all applicable ions in the B and Y ion series respectively. And if we hone in on the B ion series, we see several instances of things that we saw in our previous example. For example, that B1 ion. So that is very rarely observed in label-free spectra, but with the addition of the PSM tag label, it becomes observable in about 90% of peptide spectra. And this gain in the B ion series extends out into the higher ions as well, and so we attribute much of the enhanced score performance yielded by this tag to the stabilization of B ions. And we can also begin to look at fragment ion coverage on a per peptide basis. So there are a couple of ways we can look at this, and the first is just as a percentage of theoretical fragment ions that we're able to match with the peptide. So here I'm showing an illustrated example of a case where we have identified 6 out of 7 Y ions and 2 out of 7 B ions, and this equates to a 57% fragment ion coverage overall. If we take this metric and apply it to our set of intersected peptides from before, we see that the addition of PSM tag leads to a distinct shift toward higher percentages of theoretical fragments matched in our peptides, with a significant proportion actually showing 100% theoretical fragment matching. So this is going to be, this metric is going to have one of the highest correspondences to score in a database, traditional database search context. But in other contexts, it can make sense to look at fragment ion coverage in a different way, as coverage of fragmentation sites. So so fragmentation can occur between any pair of amino acids in the peptide, and I've marked the possible fragmentation sites with the lightning bolts. And each fragmentation site can be covered by a Y ion, a B ion, or both. But there are certain applications in which having incomplete fragment ion information is the main limiting factor. And these can include localization of post-translational modifications, and also applications like de novo sequencing, where we might be challenged to resolve the ordering of a pair of unknown amino acids in the middle of the peptide. So here I've shown an example where incomplete fragment ion coverage can still yield 100% fragmentation site coverage. And if we look at the proportion of peptides that get 100% fragmentation coverage in our proteome sample, we see that we have a huge boost to this metric as well by the addition of PSM tag. And I've broken it down by peptide length. We see that actually the greatest margin of improvement comes from peptides that are a bit longer, on the longer side. So these are traditionally very difficult to get complete fragmentation site coverage for, but with that extended B ion series that we just saw, we can have enhanced information from the N-terminal side that can flesh out the fragmentation sites in the middle. Another important characteristic of tags that we are quantifying is the retention time shifts that the tags can induce. So to set this up, in a standard tryptic digest will yield peptides that end in either lysine or arginine. In a label-free context, these do not elute significantly different from one another. Their distribution of retention times is very evenly matched. But the reason that we're interested in looking at this difference in lysine and arginine terminating peptides is that that corresponds to the number of tags that can be attached to them. So the tag will react with the N-terminus of the peptide, but it will also react with the lysine side chain. So lysine terminating peptides can bear 2 copies of the tag. And we know that having 1 copy of the tag induces a comparatively small and consistent retention time shift on the order of minutes, several minutes. But when the number of tags on the peptide increase, then this retention time shift gets compounded, which has the potential to make data analysis by various means a little bit more difficult. Unfortunately, this is not an issue for DDA approaches. So these are going to be, DDA data is going to be searched with more traditional database search software, which is traditionally, which conventionally does not leverage retention time or retention time prediction to a significant degree. So we can see this is a 2 nanogram injection of a label-free and tag sample on the Orbitrap Astral. And we see that the relative proportions of lysine and arginine terminating peptides that we're able to see in this DDA sample are relatively evenly matched. But it's a different story when it comes to DIA. So DIA search methods, conversely, rely heavily on well-matched spectral libraries, and particularly very accurate retention time prediction for every peptide in the library. And this is where we can start to see reduced performance and challenges in identifying these lysine terminating peptides. So I'm showing now 200 picogram injections on both our timsTOF SCP and Orbitrap Astral. And we can see that we have very good coverage of arginine terminating peptides on par with or better than label-free. But the lysine terminating peptides have a much lower identification rate. We attribute this mostly to suboptimal spectral libraries. So one important component of spectral library generation, which I just mentioned, was retention time prediction. So we think that the complex retention time behavior with the lysine terminating peptides makes this harder to predict, and that's one of the components of explaining this differential performance. And also it can be, we've observed that the lysine terminating peptides also have some unique spectral characteristics that might bring them less in line with the standard libraries that we use. But the good news is that despite using these suboptimal libraries, our protein identifications scale very well with plex, multiplexing. So taking the samples from the previous slide, I'm now showing the overall number of precursors and protein IDs identified in both a non-multiplex sample versus, on the left 2 panels, a PSN tag enabled 5-plex. And we see that we're able to scale from 1-plex to 5-plex with very little impact on the number of IDs obtained per plex. So this is great, and this represents an increase in multiplexing capability from what was previously attainable with a tag like MTREC. And this is also, this performance that we see from 5-plex also extends to 9-plex. So this currently using a 9-plex with the 2 Dalton spacing in tags rather than 4 Dalton spacing requires more sophisticated spectral modeling to really enable. So currently we're only showing this for PTI's own software JMod, which Kevin will be talking about next, and going into the joint modeling approach that actually enables this narrower spacing and the resultant 9-plex. But scaling looks very good for both 5 and 9-plex, and currently we hope it's just a matter of getting more accurate and complete spectral libraries to get our identifications to parity. So to summarize our work thus far, we took 576 chemical candidates that led to a mass tag that allows increased confidence of peptide sequencing. We have a 5-plex at 4 Dalton spacing and 9-plex at 2 Dalton spacing. And we have a detailed synthesis that we are publishing open source, and we're working with commercial entities for licensing and distribution. And we're also looking toward the future for the development of a generation 2 tag. So if you'll recall, our original assay was predicated on 2 synthetic peptides, and we've expanded this to a pool that we now have of 30 custom-made synthetic peptides. And we're developing an assay that will allow us to take these 30 peptides and label them with 30 or more compounds per run. And working with our partners at Creagen, we'll have 10,000 or more tag compounds to test in the near future. But with this high-throughput screening approach, this should be very doable within the coming year, and we potentially even test even more. So what we want to do with this new assay is take what we've learned from the first generation of tag development and enhance it further. So we want to select for qualities like spectral quality that we've been presenting on, see if we can maintain that and hopefully increase it further. It would also be ideal, for example, to identify tags that have a minimized or even negative retention time to shift, and that could help them work better out of the box with existing DIA software. And there are many more properties that we can quantify and leverage for the development of tags for both specific specialized and general purpose applications. And at the end of this process, we'll have a ton of data for these ton spectra accumulated for these 30 model peptides. And we hope this will help us develop a predictive model for chemical structures that enhance these features. And Mike is spearheading this effort, and he's the one who's telling me to stop talking. So you can talk to him, and he'll be very happy to give you more context on our future directions here. So I'm going to turn the mic back over to Maddy for our acknowledgement.
Lastly, we'd just like to finish with some acknowledgements, so particularly acknowledging the Mass Tags team at PTI, Sarah, Harrison, Nikolai, Keller, Mike, it was just pointed out, Jane, Corinna, Anisha, and Helenka. And then also to acknowledge our funding sources, and then also specifically shout out our collaborators at Creagen. I think Dave and Raj are here today. And then also the Pentulute Lab at MIT, who helped us a lot with the early phases of this project, and then also the rest of the members at PTI. But with that, thank you so much for listening, and we'll open the floor to some questions.
Thank you, Maddy and Mark. I'm Sarah. I'll be helping moderate questions today. I'll also be monitoring on our live stream on YouTube, so for anyone joining virtually, please do chime in with some questions, but I see we already have one here in the back, so I'm going to...
Hi, great talk, guys. The potential to screen like hundreds, well, tens of thousands of small molecules for tags in the future is really exciting. Do you have any plans, instead of kind of just the high throughput approach alone, do you have any plans to maybe train a model on the chemical structures to kind of leverage, train a model for your desired quantities, like the lack of retention time shift or fragment quality, to kind of, in addition to the high throughput method, to kind of have an intelligent design strategy?
Yeah, great question. That is precisely what we're trying to achieve. Yeah, and we're pursuing both kind of top-down and bottom-up approaches to this, so we can test like larger mass tags that are kind of variations on our existing PSN tag, and we're also interested in testing smaller building blocks that might show positive spectral improvements that could be aggregated to form larger structures as well.
Yeah, I'll just echo a beautiful talk. So I guess maybe two related questions. The first one is, it's interesting that you lose a bunch of the label-free IDs, like 8,000, some dropped out, and you picked up 8,000 new ones, so on balance it looks even. I'm just curious if you have any insights about like what are the nature of those peptides that you're losing your IDs on? And then relatedly, when you have a missed lyse cleavage, presumably you get a second tag there, and is that part of them that are going to these missed cleavages? And then finally, I was just sort of curious, like you get this nice RT shift, and I was curious whether you guys are thinking about actually taking advantage of that. You're getting some chromatographic separation, and maybe that's a feature, not a bug. Just how you're thinking about that.
Yeah, great questions. So I think the first one, yeah, we did notice that there's a pretty big difference in the intersect. This is in the context of the identities of the peptides changing between label-free and tagged, right?
Yeah, I guess I'm just surprised like there are only 4,000 PSNs that overlap.
Yeah.
And you lost 8,000 relative to level label-free, you picked up 8,000
Yeah.
relative
So we did notice that it's definitely something that we still have to explore, figuring out whether there are particular qualities of the peptides themselves that kind of lead to them being, or within the different populations of peptides identified by label-free and tagged, what the difference, physical property differences between those peptides are. We've also been wondering about what the potential biological implications of that are, but something to explore a little bit further for sure. I think I might have to ask for your help on the second question.
No, when you miss a lysine cleavage, if
Yep.
you miss cleavage there, you get an extra tag.
Yeah, so if we miss a lysine cleavage, then we'll get another tag, which I'm not even sure if we've been able to identify peptides that have had 3 tags. We've done a little bit of preliminary work with RC peptides, and what we'll find is that if we have more internal lysines, we'll just see a relative drop off in the number of peptides we can identify, and I think that might also kind of be hand in hand with the chromatographic separation. In terms of the actual usage of the retention time shift for the lysine-containing peptides with 2 small molecules on them, that's a really interesting question. I feel like right now for us that has definitely felt like a little bit more of a hindrance in terms of our DIA retention time prediction, but we'll have to think a little bit more about how that could be utilized in a helpful or innovative way.
No questions online at the moment, any?
Thank you. Just going back to that intersecting peptide Venn diagram, I'm one of the non-technical people that Nikolai is encouraging to ask questions. What's on the non-label free side? Is that just tag that hasn't attached, or what's the 8677 number?
8677.
On the Venn diagram?
Oh yeah, so this is the number of unique peptide sequences that were found to be labeled with PSM tag, but were not found to be present in the label-free peptide population.
Got it. And my second question was, did anything really surprise you or challenge you that you didn't anticipate in this set of studies?
Well, I think Mark and I will definitely have different answers to this question. Mark, do you want to go first? Okay. I would say some of the challenges that are not described, I mean, obviously we're going to present the results that are more positive results than like some of the setbacks that we've had. Something that I've been working on a lot is the approach to bulk cleanup with this, approach to bulk cleanup processes with this tag. And so I think that has been one of the challenging chapters of this work. I think going into it, I didn't have a lot of experience with bulk cleanup. And so, sorry, to give you a little bit more context, if you recall this slide we have, where we have a pretty large excess of this tag byproduct versus the amount of labeled peptides, this can cause some problems for our analysis. And so we've made some strides in particular methods that could help us mitigate this. One being ion mobility, another being changing our labeling such that we have closer to stoichiometric ratio of tag to peptides, stuff like that. But one of the other approaches that we want to be able to do is to be able to physically remove the tag from the peptides, or from the tagged peptides, in order to aid our analysis. And that has definitely been a really challenging chapter, just trying to exploit the different chemical properties of the tag relative to the tagged peptides, and figure out some creative ways to do that.
And I'll add from the data analysis side. So the tag is 308 Daltons, which is really pretty large. So it is unusual to be working with a mass addict of that large size, if we're considering one or even two copies of it. So as a lot of you have alluded to, that causes a lot of unpredictable or unpredicted maybe effects on the populations of peptides that we can identify with and without the tag. So the populations are shifted towards shorter peptides with the addition of the tag. There's also the concept of the tag having much higher hydrophobicity, which has that whole retention time shift. So there have been a lot of things like that have been maybe not foreseen in the tag design process that we've been parsing out in the data analysis side.
We are.
Am I allowed to ask you a question? Oh, there's another question.
Yes, we do have one question here, and then Nikolai has something. And
I'd like to answer Holly's question because she asked the very same. All right, I'll be very quick. So I have actually a positive and a negative surprise. So the positive surprise is we screened relatively small chemical space, and finding a tag with such favorable properties for fragmentation was better than what I expected. So I think that was a very good surprise. And that also gives me a lot of courage for screening larger space and possible improvements. The negative side has been how much effort it has been to train a good spectral library. The good part about that is it is very overcomable. It is something that currently affects the depth of our results, but this is something that we can foresee fixing relatively soon and then getting much better results.
Thanks for the talk. It's quite fascinating to hear this much about proteomics coming more from the small molecule side, MS side. One question for amino acids that have the same mass, isoleucine, leucine, how do you distinguish them? And the other, if you have, initially you were alluding to this, that you also want to see modified peptides and really look into post-translational modifications. How do you identify amino acids that have been modified and that are not the usual masses that you would expect, or even some that are new and no one has seen them before?
So a lot of that is addressed in the standard database search strategy that's really been around since like the 90s, right? So for example, your leucine isoleucine question, that is resolved for the most part by relying on a protein database that runs through a simulated digest. So we are limiting our analysis in most cases to peptides that are known sequences within the human proteome. So that will, given the context of the amino acids around that, we'll localize it to one or a few proteins, and that will kind of resolve that issue there. And what was your second part?
About modified.
Oh yeah. So in the database search context as well, basically we're limited to, in most approaches, to searching for modifications that we anticipate. For example, methionine oxidation, phosphorylation. And these can be applied, database search, we would call them dynamic modifications, where we consider both the version of the peptide that's unmodified and the version of the peptide that's modified with all the given permutations of those modifications. And whichever has the best match is the candidate that we go with.
Okay, let's thank Maddy and Mark one more time. And I invite Casey here to introduce our next speaker.
Hello everyone. My name is Casey Laguna, and I am both a student here at Northeastern and a current computational co-op at PTI, where I work under Dr. Kevin McDonnell, who I'm here to introduce. Kevin is a senior computational scientist at PTI who develops analysis pipelines and new peptide identification tools for proteomics, which is what he will be speaking to you all about today. Kevin received a bachelor's in theoretical physics and a master's in data analytics from the University of Galway, where he stayed and continued his studies to complete his PhD in machine learning techniques for de novo peptide identification. It has been an honor to work with and learn from Kevin as a very supportive, informative, and talented scientist, which is why I am happy to introduce both him and his presentation on JMod, Joint Modeling of Mass Spectra for Empowering Multiplexed DIA Proteomics.
Yes, thank you very much for that introduction, Casey. Okay, perfect. I'm going to talk about joint modeling of mass spectra for empowering multiplexed DIA proteomics. We've just heard about the real opportunities and capabilities that PSM tag offers in terms of the number of parallel samples we can analyze together. But I'm going to talk a bit about how we actually go about searching that kind of data. What are the challenges that this kind of multiplexing creates, but what are the actual structures within the data that we can utilize that we can help search this kind of data. So once again, we refer to our pyramid. And like was said previously, we can typically parallelize a number of samples. We might do this in a DDA context, where we can get up to 35 plex. But this is again, as was said, sampling one peptide at a time. We could parallelize the number of peptides in the DIA context, where we can sample multiple peptides per scan. But the level of multiplexing in this domain has been somewhat limited to date. But now with PSM tag, we can start to scale here. So as we multiplex in this domain, we are going to sample all these peptides, but we have to make sure we still identify them. So as we increase and climb this pyramid, we don't want to sacrifice the number of peptides we're identifying at proteomic depth, as was talked about. As we increase the number of samples, we want to maintain both of these to keep our equation of throughput true. So just to give a slight bit of background, this is probably known to a lot of people here, but just to get everyone up to speed. In bottom-up proteomics, we typically couple our mass spectrometer to an LC system, which separates our peptides in time. And then our mass spectrometer will separate them in the mass and charge domain. And so we get this ion map of peptides, different molecules that are separated in mass and time. And what we want to do is we want to sample these, as was said previously, we sample a section of these fragments, or sample of these ions, and we send them to fragmentation, and we can do identification that way. And just to talk about these little funny shapes we see, these parallel lines, well, these are actually the isotopic envelopes we see. And I want to talk about this because we don't isolate a single ion for every peptide, particularly in DIA mass spectrometry. We sample the full isotopic envelope, and this will become pretty relevant later on. So the different ways we sample this ion map, as I said, we can do it a data-dependent acquisition method, where we take a survey MS1 scan, and then based on the ions we see, we can then isolate those and send those for fragmentation. But you can see how this is a pretty sparse sampling of our full ion map. And we want to, there are alternative approaches like data-independent acquisition, which gave us a greater sampling. So we again take this MS1 scan, and then we have repetitive cycles of these windows in every duty cycle. And you can see the relative size of these windows is much larger. So we're sampling more ions, potentially multiple peptides per scan. We also have the additional dimension that we sample the same peptide multiple times. So we get this, we can capture it as it elutes off the column at different points in time. But because we have multiple peptides in a single spectrum, it's not able to it's not simple to disentangle what fragments came from which. So in the examples that were shown previously, there was a DDA spectrum where all the fragments are assumed to come from a single peptide. What happens and how do we search data when that is not the case? So this is the case, cartoon version of what was shown previously. We observe some fragments, some peaks in M over Z space, and we have a library of peptides we've seen before, and we want to assign with some level of confidence, okay, this looks similar to this one we've seen before, and with some level of confidence say, okay, we've identified this peptide. In a data-independent acquisition scheme, it become more complex. So we have multiple peptides. Potentially, all of these can be present, but how do we disentangle this mess that we see? Some peptides, as we can see, have a single source. We're not sure which source they came from. We can see here in this cartoon, but these are not labeled when they come off the mass spectrometer. But the most important case here is some fragments can come from multiple sources. So they can be a superposition of contributions from the different peptides that were co-eluded. And we can look at the rate of this, so how many peaks are considered distinct for each peptide, each precursor. And when I say precursor, I should note, to limit the jargon, when I say precursor, it's a peptide with a given charge state. And so what fraction of those fragments are distinct for a given peptide? And we do this with in silico with a set of observed peptides we've seen previously, about 70,000. And we look at what fragments are distinct, and we see that most fragments for most peptides are actually not distinct. And this is a label-free DIA experiment. These are 16 Thomson-wide windows. These are 16 Thomson-wide windows, so not that large. We can see as we increase the size of these windows, the number of distinct peaks actually goes down. And you can say, well, why do we care if we increase the size of our windows? Well, in a single cell proteomics example, maybe we want to capture more of the ions. So with smaller windows, you actually throw away most of your ions. In the larger window, you can actually capture more of the greater fraction of the ions. Or you could actually increase your sampling rate. So you could, with larger windows, you may want to sample your ion or your elution peak more frequently, and that's afforded by using larger windows. And those window sizes are just based on dividing this mass range into these sizes. So we have an exponential increase, and we see that decrease. So, okay, many peaks are distinct, this is label free. But here at PTI we're interested in multiplexing. We want to scale this up. We don't want to deal with label free, we want to increase the throughput. So if we were to multiplex with non-isobaric tags, this is the effect on these overlapping peaks. So we get even fewer and fewer distinct peaks as we go from a 3-plex, label-free, to a 3-plex DIA, to a 6-plex DIA. And so this level of overlap is inevitable as we increase multiplexing. And so what we want to ask the question is, how can we handle these data? What is the best methodologies we can set up that we can deal with this data and this inevitable increase in overlap? So there are different ways we can deal with this. We could throw out overlapping signal. Of course, this is not ideal, given that we'll be left with very few as we multiplex to even greater amounts. We could assign to a single precursor through competition. So if a fragment is potentially shared, maybe we only wanted to give it to use it to assign confidence to an individual precursor. Or maybe we can just model it. So we can accept this overlap happens, it's going to increase, and so let's try and deal with it. Let's take the data for what it is and try and model it. So this is the strategy we decided to use as we develop our software here. So what we're doing here at PTI is we're developing this framework. We're calling it JMod, standing for Joint Modeling. of DIA spectra. And the goal of this framework is to support multiplex. We want to enable increased throughput. I'm going to talk to you today about one method of that in the terms of as we multiplex in the mass domain. But this framework can work for any type of multiplexing, and there are other types you'll hear about later and we are pursuing as well. And also we want it to be open source. So we want people that are using this, researchers, how are your data generated? We want you to be able to see the code, see the software, how it works and how it's generating your data, and the people can collaborate. So maybe there's a research group out there that can sort of use this as a foundation to address the challenges that are unique to their lab. And we are open and welcome collaboration if people are interested in doing so also. So just to talk about this a bit more, in terms of this is conceptually what the problem is. We have this superposition of fragments and a set of library spectra that compose it. How do we go about disentangling this? So what we can do is we can set up our equation here. We start off with our observed spectrum. So we have our matched peaks as a list of observed intensities. So we can just, we don't need to worry about M over Z. M over Z can be done for matching, but so we have a list of observed intensities that we want to model. And in our library, we ideally have all possible peptides or all possible spectra that could be contributing to this signal. And so what we do is we just fill in the columns of this matrix with our observed spectra, and each row here will correspond to the, each row here is a fragment in our library that is matched to this particular observed peak. If it does not match, we can just fill it with a 0. And the question is, can we find some set of coefficients that balance this equation? When we multiply our library intensities by this set of coefficients, can we get back our observed spectrum? So essentially, you'll be doing, if you remember your matrix or your linear algebra, each row by column. So this will end up all be multiplied by the single coefficient. And so what we get back is if we can find the set of coefficients, is this particular one will give you the abundance of this library spectrum in our observed spectrum. And we can do this for all the independent and individual spectra that we observe off the mass spectrometer. And so we get a set of coefficients for each of these particular peptides, and we can trace out their elution curve and their abundances in the spectrum. And so if we want to find the set of coefficients, we just want to minimize the residuals. So multiplying these two together will give us a vector, and we can minimize the residuals between this side of the equation and this side of the equation and find the set of coefficients that does so. So let's look at a real example. So here, to simplify it and make it observable, I'm looking at the monoisotopic peaks. So no isotopes here. Of observed peaks matched to these two very similar peptides. These two peptides differ by one amino acid, and as was alluded to earlier, then they will share a lot of the same fragments. And so you can see here, using this joint modeling approach, we can deconvolve the signal, and so we can see we can accurately attribute the intensities to individual fragments, so fragments that are just attributable to one peptide. But importantly here, we can model this superposition of fragment intensities that happen when both peptides contribute to an ion. But this is label free. This approach is used by others. It's not unique to us. Spectre, Chimeras, Pioneer all use this approach. What we want to do is we want to utilize this in terms of multiplexing. As we increase this level of overlap, how can we use this to get still accurate quantitation and identification? So we started evaluating with PSN tag. So as you've seen before, this is PSN tag here on the left. And all the dopable sites you can see where ions or isotopes can be added. And that gives us these 9 possible configurations of the tag. And so this evaluation we can do is we can look at this in context of a single channel. If we only treat a sample using just the delta 0, the base tag. Or we can set up this 5-plex example. So as was alluded to earlier as well, typically in DIA mass spectrometry, when we're multiplexing, we ensure that there are 4 Daltons between each channel, each sample. And this is to account for the isotopic overlap. It makes our lives a lot simpler, and most algorithms can work with the data if there is little to no overlap between these channels. And 4 Dalton achieves that. But if we were to relax that constraint and we were to able to develop models that could, even if we could just relax that to 2 Daltons and develop models, we would almost double the effective throughput we could get for the same molecule. So this is where joint modeling comes in. We start off with this 5-plex example. So you can see here, the different isotopic envelopes. This is the observed MS1 intensities for this particular peptide. And you can see with joint modeling, we can get back these intensities by finding coefficients at the MS1 level now that match the expected or theoretical isotopic envelope to the observed data in the same way. But in the case of our 9-plex, now we don't have discernible differences in the isotopic envelope. And so we rely on this joint modeling approach, but we can get back the individual contributions from each of the different channels, each of the different samples. And so in a biological context, these would be peptides from different samples. And what we can do is we can get back the individual contributions from each sample, thereby quantify the peptide distinctly in each sample by deconvoluting these beads. So in the context of the number of identifications we can get, so this is the number of protein IDs we are getting for a 200 picogram sample on the Orbitrap Astral. So the Astral, 200 picograms is typically how we use to model a single cell level input. It's pretty low. And you can see for label free, we're getting over 4,000 proteins, slightly fewer for the delta zero. But what's important here is that we are scaling. So we're not, when you remember that pyramid, as we increase the throughput, as we increase the number of samples we are analyzing parallel, we are not sacrificing the level of depth for this particular tag that we're doing. We are scaling multiplicatively with the number of proteins. And so to compare this to Diane for the same data, Diane is getting about 5,000 IDs and about 4,000 IDs or fewer for delta zero on the 5-plex. But important here, Diane or no other search engine can search this 9-plex data. So JMod is unique in its current abilities to search this data. So no other search algorithm can give you this level of throughput for this data. And so IDs are not everything. We also care about quantitation. It's very important that if you observe a peptide or a protein, how abundant is it relative to the other samples. And so to do this, we set up a mixed species where we mix human and yeast proteomes together. This is typical in the proteomics community. You could say it's similar to a spiking, where you have known input amounts of human where in each mixture. We have 200 picograms in A through D. But in yeast, we have varying amounts where we have from little as 20 picograms up to 80 picograms in these samples. So that gives us 4 different mixtures we can use. With PSM tag, we have 9 different configurations for our tag, 0 through 16, every second one. And so we just label each of the different mixtures with these different tags, with these different configurations. And these are then mixed into one, run through a mass spectrometer, and so we can search the data and try and evaluate, can we get back these known ratios once we search the data. And so I've shown you a 5-plex peptide before. This was human, obviously because we can see the similar abundances for each of the 5 channels. But if you have a yeast peptide, you can see how this changes dramatically and how now the contributions from a fairly abundant channel can actually significantly contribute to the abundances of the delta 12 channel in this case. And so this is where joint modeling becomes so important. This case is a particularly hard example, but it just shows how joint modeling can be applicable even to the 4 Dalton space, which is typical for DIA multiplexing. And so what we can do is we can take all of the delta zeros and all of the delta 12s and compare them. They should have a 4-to-1 ratio. And on a log scale, you can see how the yeast peptides in this context all get accurate quantitation, or not all accurate, but there is accurate quantitation for the majority of these yeast peptides, and humans are centered at 0, so on a log scale they have a 1-to-1 ratio. We can do this more systematically, so we can look at all the different mixture levels and collapse them, so all the A over B's in this data, and compare that to the theoretical mixing ratios. And we can do this for all of the different combinations. So now we have all the different channels and their mixtures shown here on the data for the 5-plex 4 Dalton, and we can compare that to Diane. So Diane, as I said, is the leading search engine in this space. And you can see how we're getting similar, if not better, quant for these are intersected peptides. So there's no, these are the same peptides identified in the Spiplex experiment between both JMod and Diane, and we're getting similar, if not better, quantitative accuracy. But of course, that's with 4 Dalton offsets. What about the case when we have much greater overlap? So I showed you, as I said previously, the human example. This was a significant signal in each of our channels. We can deconvolve this. But you can see how challenges may appear at first when you have a yeast peptide, and you can see how the different input amounts, again, if we refer back to delta 12, it never contributes more than half of the signal to any of the ions it's matched to. And so we can evaluate the quantitative performance of the 9-plex. And so here I'm comparing it to a 5-plex, this is for using JMod, the 4 Dalton offsets, and the 9-plex with the 2 Dalton offsets. And you can see for these examples, we're getting quantitative accuracy centered around the expected mixing ratios. And it's pretty comparable between both. So we've halved the distance, we've doubled the overlap, if not more, and we've doubled our throughput, yet we maintain quantitative accuracy in these data. And so we can focus in on these two particular examples. So this is the 4:1, this is the largest ratio we have in this particular data we've example. And you can see a direct comparison where they're comparable quantitative accuracy between both methods. And to zoom in on those two again, so we look again at our delta 0 versus delta 12 here for the 5-plex, and the delta 0 versus delta 12 for the 9-plex. And you can see how they're pretty comparable in terms of the accuracy of quantitation. But just to refer back to what's actually happening here, so this delta 12 is being pulled out of this signal. So really powerful, this joint modeling approach, how it can actually get accurate signal from this, and thereby double almost effectively the throughput we can get for the same molecule. So I've talked about PSM tag, I've talked about the Astral, but what about other approaches? So there are alternative tags such as diethylation, where we add ethyl groups to the primary amines of our amino acids, or our peptides, excuse me. And depending on the reagents you have available, this can achieve a 6 to 8-plex with 2 Dalton offsets if using trypsin. This has been done before with DDA. It has never been done or utilized with DIA before. But JMod and its joint modeling has this natural ability to deal with this data, similar as I've shown. One of the things to know with diethylation is to get this level of multiplexing, one must use deuterium. Deuterium affects chromatography and therefore retention times. And so while with PSM tag, you will see the different channels, the different samples loop together at the same time, and it makes quantitation identification easier. Deuterium offsets this so you don't actually see, the channels don't elute off the column at the exact same time. However, acquiring this data, again 200 picograms, really low input level amounts, on an Explorers 480, so it's now not your state-of-the-art Astral, it's a different instrument, more affordable standard instrument, and we can still achieve accurate quantitation. So this is a 6-plex for diethylation with 2 Dalton offsets, and we still maintain quantitative accuracy. As was noted previously, I guess something to acknowledge is the accuracy of this problem is highly dependent on your library. Because if you want to minimize the residuals, as we've talked about, between this side of the equation and this side of the equation, you need an accurate representation of your library. And this not only includes having the correct fragment intensities, but having all the possible library entries present also. And this is something, while if you look at a label-free example, we can say, well, how similar were these both sides of the equation? In label-free, we can see they're very highly similar. So this is a measure of how well we predicted each spectrum we observed. Currently, it's slightly worse for PSM tag, but as Nikolai said in the previous questions, this is something we can address. So these label-free models have been trained on millions and millions of spectra. We are just acquiring that data. We are optimizing these libraries all the time. So the results I've shown you will inevitably get better as we get a better model of our data and therefore better library predictions. So just to conclude, JMod enables scalable proteome depth with large plex size. So as we increase the number of samples we run in parallel, we're not sacrificing the number of proteins we see. It provides accurate quantitation for single cell level inputs. So these really low level inputs, we can still get back the expected quantities we want. And it affords increased throughput. So I talked today about one methodology where we use small mass offsets, where we can almost double the throughput we can get for similar molecules. But Jason's going to follow this talk talking about another dimension we can increase throughput that is also enabled by this joint modeling approach. And so to finally just acknowledge again the people that made this happen, the whole team at PTI, who are an absolute pleasure to work with. I want to make notable note of Sarah and Maddy, who have been great in terms of helping acquire the data and giving advice in general. And of course, Jason and Nathan, who have made extensive contributions to JMod and have been excellent to work with. And so thank you for listening. I guess to finish on, this is on GitHub. It will be publicly available in the next week or so. It'll be at this handle. If you want, please download it, try it out, let us know what you think. You can contact us through the GitHub, or if you want to contact me, my email is there, and you can get in touch if you wish to collaborate. Thank you.
Thanks, Kevin. Let's open it up to some questions. I'll start with one. So we've seen 4 Dalton spacing, we've seen 2 Dalton spacing. What are the limitations to going smaller, like single Dalton offsets?
This is something we've explored with non-commercially available tags previously, and for the analysis we did at the time was only on bulk, and we did get accurate quantitation. So we believe it is possible. Again, I think one of the main limitations, you need accurate representations in your library once you go to that. We have a couple of improvements we have still to make, as Nikolai alluded to. This is still a work in progress. This is version 1 of JMod and will be improved better, but theoretically there's no limitation. What you run into is if you don't have everything in your spectrum. So you have some type of peptide that is not in your library, and maybe that causes an interference in one of your channels. And now that you're at single Dalton spacing, maybe that becomes a bigger issue. But we don't see there's a huge issue with single Dalton. That's something we're going to actually explore in the next few weeks also.
No question.
Hi, Kevin, great talk. So from my understanding, the quantitation is on the MS1 monoisotopic piece.
What we actually take is at the start about MS2 deconvolution, but we can do the exact same principles at MS1. If I can go back. Yeah, so we can take this example. So for this is a particular MS1 spectrum. And so this joint modeling will give us a coefficient for each of these channels in this particular spectrum. And what we can do is we can do that for every single MS1 spectrum, and what you get is a series of coefficients that trace out the abundance of the peptide over time. And we just take the area underneath those coefficients as our quantity.
Right. So for the coefficients, have you considered adding in external factors like the peptide length into consideration?
We take that into account in terms of so what we're actually, I should say we're fitting here is the theoretical isotopic envelope. So that will inevitably take into the peptide length because of the number of carbon 13s we have. So again, having an accurate model of that is essential for this. So that was something that's actually interesting. You bring that up. We're dealing with this is we have to take in the tags we have currently developed have their own isotopic envelope. And because they're quite large, that is significant. And so we have to actually account for that in this modeling approach because it's significant.
Got it, thank you.
Yeah, some here in the back.
Hey, Kevin. Thanks for the talk. So you mentioned the tags have their own envelope. Do you have a rough idea about the purity of the tags?
The purity of the tags?
Or the isotopic purity in terms of...
I believe it's pretty high. It's not a question I can answer. Maybe someone else can answer. But their distribution they have a natural distribution. So I guess maybe what you're getting to is as we actually remove some of the dopable sites, the natural distribution will actually differ for the delta 16 versus the delta 0 because there are fewable natural positions that carbon 13 can hold. So in terms of purity, I think it's pretty high in terms of the actual, when we dope the sites. but then sites that are undoped still have this distribution. So we just say, okay, what is the number of sites that are undoped? What would be the natural distribution based on the presence of carbon-13 values?
And have you found that for the JMod workflow that the narrow window, like Astral DIA, has that been a big boost in terms of the performance that you've been seeing? Or does it, even without that fast MS2 scanning capability?
Yeah, so narrow window is an interesting example. With this Astral data, I don't have but I think it's about 20 windows, so we're not utilizing the tiny windows that can be afforded by the Astral. But one thing to note with the Astral, say at the MS2 level, is you may not be capturing the full isotopic envelope, and this is something Nathan is working with us has discussed and we plans to implement. So if you capture only a fraction of the isotopic envelope of the precursor, and then you send that for fragmentation, that will affect the fragmentation isotopic envelopes. And this is something we need to model. So we actually include isotopes at the MS2 level because those are peaks we want to see. So we need to account for, okay, if we only capture part of the envelope, whether it's the start or the end, how will that impact the fragmentation isotopic envelope? So that's something we can model and predict.
Thank you.
This is going to be a basic question. In the presentation before and also in this presentation, you guys kind of talked about the difficulties with spectral libraries. Can you touch a little bit on what steps you're taking or what experiments you guys are doing to optimize those spectral libraries?
Yeah, cool. So in terms of the label-free libraries I'm using, I use ProZit, trained on hundreds, thousands, maybe millions of spectra of library-free data. There are different approaches you can train from scratch. And so we have these really large molecules, as you saw, affect chromatography. They also affect fragmentation. And so we could take a model that's already trained to predict a label-free data and fine-tune that to these to the changes we observe due to this tag. Or we could train from scratch. We currently don't have a lot of data or enough data to train from scratch, so we've been working with fine-tuning from a label-free model or from an in-track trained model. But the difficulty is, I think, that this tag, because it's so different to these others, the models struggle to actually get to that place where it's accurate. So we're currently seeing, as I showed, we can't optimize it fully, even though we have thousands of spectra, it's simply not enough. So we're currently thinking of different ways to frame the problem in terms of how do we include the tag in these prediction models. So that's one of the ways we're exploring, and how can we better fine-tune these models. But yeah, it's an open question.
Maybe really quick, I know Silex sort of out of vogue with all the tagging and high multiplexing, but if you looked at the quantitative accuracy, if you just had like a 2-channel Silac, do you have a sense of how tight the distribution would be?
We did experiments, I don't have them here, no, I don't have them, where we looked at the delta 0 and delta 2 versus delta 0 and delta 4 only. So a similar example, so it's not the effect of multiplexing. And quantitative accuracy is pretty similar. So not only have you, I guess, with the 9-plex you've increased the number of peaks, well, the complexity of your samples, and so that makes fitting a bit more difficult. But if you were to just look at the two together, it's actually comparable without the effect of increased multiplexing.
Any more questions from the audience?
Have you evaluated the resolution, how the resolution impact the quantitative accuracy? And for example, on the Astral, you have a very high resolution mode versus timestamp, or if the ion mobility additional dimensional separation can help you to reduce the overlapping pattern. These kind of factor, how they impact the accuracy of the quantity.
Yeah, so on the basis of resolution, yeah, we do see greater quantitative accuracy on the Astral. This modeling approach is really good if you have this shared fragment. So if you've made the same ion because of the exact same partial peptide sequence, so the exact same fragment, they'll be identical in overseas. But if you have interferences now from similar peaks that are not resolvable, you have a very high peak next to a very small, that is not something, that's just a factor the mass of the mass spec resolution, and not something we can model correctly, because we just get centroided peaks here. That's what we model. Sorry, what was the second part of the question?
The separation, either the gas
Oh, and
separation
sorry.
or the LC algorithm reduce the closure, and how do you get better on it?
Yeah, I don't have that data on hand. We have explored using this on timsTOF SCP. We haven't a good method of doing that. It works, but we're reliant on other software to centroid the data, which hasn't been working the best in our hands, but it's something we're actively working on. So we have this joint modeling approach working. We don't have it evaluated in this context on the ion mobility separated data. But if you can set up your problem that you're still just fitting those peaks, it's fine in that context.
Any other questions from the audience? Oh, we have one online. Okay, let me read this. How, sorry. I believe there's a missing word. How do these tags behave for PTM analysis, and will there be additional complexity for data analysis?
It's not something we've explored, but something we really want to look at now. As we kind of finish this 2 years, this initial phase 1, is I can't really comment on the tags in terms of this additional complexity, but this joint modeling approach, as you can see, for very similar peptides, which may be PTMs, they may, if we have wider windows, they may end up in the same spectrum. And so this joint modeling approach naturally affords us up to dealing with that kinds of data. So it's something we're excited to explore, we want to look more into and how it works, but it's not something I have data on yet, I guess, and I can't comment on how the tag will affect that.
Any other questions we have for Kevin and joint modeling? All right, well then let's give another round of applause. And next I'll welcome Arturo to introduce our next speaker.
Hello there, everyone. Thank you all for coming. It is my pleasure to introduce our next speaker, Jason Derks. Jason earned his PhD in Professor Nikolai Slavov's lab at Northeastern in 2022. His doctoral work focused on increasing the throughput of bulk and single-cell proteomics through the development of multiplexed data-independent acquisition, also known as multiplexed DIA. As a postdoc in the same lab, Jason applied these principles to quantify the proteomes of thousands of single nuclei to link proteomic and functional variability for identifying regulators of protein transport. Since joining PTI, Jason has worked on developing a new multiplexing approach to further scale throughput of LC-MS proteomics, which we'll hear about now. Please welcome Jason.
Thank you for the introduction. I have a lot of high resolution images, and that's why it's going to take a little while to load.
Is there any way to increase the audio, like for the stream? A couple people have commented that it's
Yeah. It'll load.
I don't think there's much else I can I have turned it up a bit more.
Yeah, so thanks for the
I think it just kind of depends on the person.
So yeah, we've heard in the talks preceding this of multiplexing proteomic analysis in this mass domain. And what I will discuss now, this loads, will be multiplexing in this orthogonal domain in time. Great, yeah. So very excited to introduce this for the first time publicly. I think this is really interesting technology that we've been developing to scale throughput for analysis. So why would we want to increase the throughput of proteomics analysis? The reason would be to generate large-scale data, such as maybe analyze large cohort analysis. There's many covariates that come along with analyzing pathologies that you would want to investigate the results and the causes of the covariation of health status, sex, age, things like this. The pathologies, by generating large-scale data, you might be able to investigate this. So if we had this goal of analyzing 1 million samples, this really ambitious goal, by today's standards of what most people would consider high-throughput proteomics, it would be maybe 100 samples per day. And if we wanted to analyze this, it would take many decades and cost many tens of millions of dollars to do this. So technically, very impractical and infeasible. So there is this really strong need, urgent need, to increase the throughput of proteomic analysis. And so how can we do this? We can do this really simply by just increasing the rate of data acquisition. We can churn through samples a lot faster. So we can shorten LC separations, for example. This is work done by Messer from the Rausser lab, where they used an EvoSep system to get down to less than even a minute long gradients. So they're getting really fast separations to enable more runs per unit time. This does come with the downside that people are mostly familiar with, that as you reduce your separations, you're going to likewise reduce your coverage per unit time. And so there's other ways that you might want to go about multiplexing or go about increasing throughput. Another way would be to just make more efficient use of the time that you spend analyzing samples. This is work by Shinol from the Brian Kelly lab, where they have 2 columns. One is undergoing all the overheads associated with sample loading, column equilibration, and the other is actively separating chemical analytes. And so you can put all your overheads on one column, while another column is actually doing the things that you want it to do. And you can do this switching back and forth, and that makes more efficient use of this technology. Another way would be multiplexing samples. So these approaches are all complementary. You can do both or one. Today, the talk is going to focus on multiplexing. And a common way of achieving this would be to do isobaric labeling, in which case you'll have, maybe you'll use TMT and you'll have a 35-plex, for example. These are going to be, once you pull them together, they're going to have like this superposition that's observable at the MS1 level. You can fragment these charged peptides and you'll get sequence information, and you'll get quantitation that is specific to each individual sample. This has really high sample multiplexing capacity with a 35-plex, but the coverage that you get per unit time has a ceiling on the number of MS2 scans that you can possibly take. So we can really scale potentially the sample throughput, but the coverage that you get per unit time is going to come at a detriment. Another technology that's been growing in popularity a lot over the last 5-10 years or so is data-independent acquisition. This is a way of acquiring data that will intentionally generate chimeric spectra so that you can quantify dozens or hundreds of peptides in a single scan. And this allows you to get really deep proteomic coverage very quickly. And so a few years ago, we developed this capacity to enable multiplexed data-independent acquisition so that you can get deep proteomic coverage while also having this multiplexing effect. And the benefit of this is that you'll increase your throughput multiplicatively, and then as a result of this, because all the costs are associated with LC-MS time, by increasing throughput, you also reduce the costs. So you get this dual benefit effect. Now, what I'm really excited to talk about today is this new capacity of multiplexing samples in this orthogonal dimension. So rather than multiplexing samples in this mass domain, potentially we can multiplex samples with time offsets. So we can temporarily encode samples to undergo the exact same separations and just stagger them in time. And so in this way, the time offsets would be orthogonal, or would basically be orthogonal to mass offsets. To give you a more intuitive view of how this might look, we can start with an example that I think most of us maybe are familiar with, where you have samples that you're going to load onto a column. You're peptides will bind this column, and then you'll flow increasing amounts of organic solvent over that column, and you'll have peptides that elute in a way that is highly predictable and repeatable. And so this is being illustrated here with peptide I, that is red, followed by yellow, green, and so on. And the idea with timeplex is that if we stagger in time the introduction of this gradient, then all of these peptides are going to be separated by this systematic time offset that is going to be held constant throughout all these peptides. And so now, rather than encoding samples in the mass domain, they're encoded by time separations that are systematically held. They're going to undergo the exact same separations. In this way, we can encode this time differential by just having transfer lines that contain different volumes. As you flow at a given flow rate, they're going to have travel times that are effectively different, and that's how you can encode very simply this time differential. And potentially you can do this for as many timeplexes as is practically feasible. And today I'll illustrate a 3-plex. Now what's really, I think, very cool about this approach is that it's orthogonal to the mass dimension, which actually means that you can do both. You can multiplex in this mass dimension with plex DIA, and you can multiplex in the time dimension with timeplex. And so if effectively you have a 3-plex DIA, and you have a 3-timeplex, that effectively gives you a 9-plex. So you can actually quadratically scale the throughput that you would get, and also the cost savings. So any developments that we make on the plex DI side with developing PSM tag, 9-plex, and in the future, it's all going to contribute synergistically with timeplex, and we'll get this really rapid, accelerated increase in throughput and cost savings. And so I was really fortunate to have Kevin talk before me. So how do we go about actually analyzing this data? And it ends up being very similar to exactly what Kevin had said. The only difference now is that we're going to need defined retention times, expectations of when a peptide is going to elute for a given sample. As long as we can have that, then we're going to have sample specificity for each peptide. We're going to know each peptide originated from a given sample, and we can, when we do the search, we can basically just set thresholds on when we'll consider a peptide to map to a given sample. In which case, what I'm showing here is real data. This is the result of a fine-tuning that we have done and integrated automatically into JMod, where I'm highlighting in this magenta color a particular charged peptide, a precursor. The data doesn't always look like that. You might have a library. Library is basically just an expectation of when a peptide is going to elute. Maybe your expectations for when a peptide is going to elute are really off, like very wrong. In which case, we're going to have a really hard time figuring out what peptide originated from what sample. This is actually a solvable problem. This is not something a result of timeplex not necessarily working. All of the error that we're seeing, all the jitter we're seeing, is a result of the library retention time not accurately reflecting the separation that these actually have. And so we did a lot of work on implementing a flexible solution to solving this problem, which is getting accurate retention times for peptide elutions. And really happy to discuss this more in detail. For the sake of time, I'll keep it brief and just say, basically we've trained a model. We have our own custom predictor for retention times that we then use transfer learning on based on a subset of initial identifications that we get. And we adjust all of our expectations in this library now, so we can go basically from this left-hand side panel to the right-hand side panel, where we have really rigorous expectations, strong expectations for when peptides should elute, and we can get basically completely unambiguous identifications for which peptides belong to what sample. And happy to answer more questions in more detail about how we actually do that in the Q&A. So how well does this actually work? We have this way, potentially, of analyzing these samples. We wanted to benchmark, are we able to get sample specificity with timeplex? So to do this, we went to our trusty mixed species benchmarks. We have timeplexes that are offset by 4 minutes in this case. All these samples, all 3 of these samples are going to have human. And then the last 2 samples are going to have yeast. So it's going to be missing yeast in the first sample, and then we have this plant proteome. It's not going to be present in any of these. And we're going to search this data with a spectral library, FASTAs, that basically map to human, yeast, and Arabidopsis. And what we should see is that we have good human identifications across all the different samples. We lack yeast identifications in the first sample, and we don't have any Arabidopsis identifications in any of our samples. And so here what I'm showing is the raw data from this time-plex run. And so what I'm showing here at the bottom is the total extracted ion chromatogram, and we have gap TH, a precursor that corresponds to gap TH, and I'm plotting intensities for a precursor for each one of the 3 time channels. So we have good appreciable signal in all 3 of the time channels for the human, and for the yeast, there's this absence of signal in our first time channel, suggesting that we're getting good FDR at least for these 2 peptides. So how does it look for the rest of the proteome? So if we do a more systematic analysis, we're getting about 20,000 or so identifications of human, a large number of identifications for human in all of the time channels. And I'm plotting here the intensities on the x-axis. And we have very low identifications for yeast in the first time channel as expected, and very low identifications for Arabidopsis in all the time channels as well, suggesting that we have good false discovery rate control. Okay, so to summarize this analysis, we have this potential to multiplex samples in this time domain that's orthogonal to mass. And this time offset is really analogous to mass offsets. So time offsets in timeplex are analogous to mass offsets in plex DIA. We can leverage this almost as equally well as you can in timeplex, but the requirement to do that is that you have accurate retention time predictions so that you can get sample specificity. And so we showed that we have this approach of searching this data that can give us good control of false discovery rates, shown here. And so now we want to benchmark the performance of timeplex relative to more established methods like label-free DIA and plex DIA. And we've also done this combinatorial timeplex and plex DIA, and we want to see how well this stacks up to these established methods. So to do this, we had a sample, which we're going to call A, B, and C. They had 8 nanograms of human protein lysate in all 3 of the samples, and then a variable spike in of yeast so that we can benchmark the quantitative accuracy. And to show the generality of this implementation, that timeplex can basically be implemented in many different instantiations. We have 2 different implementations that I will show today. The first of which, we have 3 columns. They're going to be loaded, and then as the solvent flows over them, they're going to be joined and then sprayed from a single emitter tip. The other one is going to have separate emitters throughout using ion optics columns. So 2 different implementations. So what do we see when we get this data? The current implementations are good proof of principle. They work reasonably well. What we do observe is that there is this drop in intensity, which is likely the result of increased ion plume density as you spray multiplicatively greater solvent at the mass spectrometer. There's going to be reduced desolvation, which likely is the result of what we see here, where we have basically one sample that's loaded, spraying at the mass spectrometer with the 1 column, or another sample that's loaded, spraying at the mass spectrometer with 2 columns that are background spraying. So we have about 2-fold drop in intensities with this current implementation, and about a 20% drop in protein identifications. This drop is probably going to be less significant as you go to higher and higher protein amounts, because a 2-fold drop at 10 nanograms on an Explorer's 240 actually ends up being pretty significant versus like injecting 10 times that amount. So this is the implementation we're going to use. We think that it can definitely improve in the future, and we wanted to investigate how well does the data type itself stack up to these other implementations. Assuming that ourselves or others in the community are going to improve this implementation, how well does the data type actually look and stack up? So we're going to control for the implementation so we can get a look at what the data actually looks like. So what I'm showing here are the number of proteins that are sample specific that are identified per run. And so what we see here is that we're getting about almost close to a threefold scaling of plex DI and the label-free DI, so we're getting proportional scaling, so we're getting about threefold more proteins per run that are sample specific for the 3 plexes. And then for the combinatorial 3 plex DI, 3 time plex, we're getting about 6x the throughput, give or take, in terms of throughput defined as number of proteins identified per unit time. And so yeah, we're getting proportionally about 2/3 coverage, which we think that, and I'll present at the end, we have some evidence to suggest that this can be improved many different ways, but one of which is analytically with spectral libraries, which Kevin hinted at earlier. We have really similar trends in the other implementation that we have. So this is the single emitter join flow, very similar trends for both of these. So the next thing we did was we looked at the quantitative accuracy of this time plex. And the first thing that struck us is that there is these changes in relative ion intensities as a function of the retention time. This is common if you have samples that you're running on multiple different columns, and for example, maybe you need to, maybe one column there's a batch effect, and you need to basically correct for this. This is something that most search engines will already handle, is this normalization as a function of retention time. We're able to, so basically what it is, we normalize for these relative intensities as a function of the retention time, just fit a lowest regression and fit that basically to 0, and then that basically solves a lot of our issue with quantitation. And so now we're able to basically get quantitative accuracy that reflects the mixing ratios that we would have expected from this given spike count. Okay, so this is showing the quantitative accuracy for the separate emitters approach and also for the single emitter approach, suggesting both implementations give good quantitative accuracy. All right, so now we're going to compare the different methods, the label-free no-plex and plex DI to these other methods. So these are the established methods that we have. Plex DI is going to increase sample throughput threefold multiplicatively. We observe that we have very good quantitative accuracy, even by multiplexing threefold. This is what we observed a few years ago as well. And with time-plex, we're also getting this scaling of sample throughput by threefold and maintaining good quantitative accuracy. Now, if we go to the combinatorial 3 plex DI, 3 time-plex, the median accuracy remains the same, albeit the spread does increase. And we are increasing sample throughput by 9-fold in this case. So to summarize the benchmarking, time-plex can be implemented in many different ways, some of which we are hopeful that will improve in the very near future. We're getting this about 2/3 proportional scaling of coverage. We think that we can improve this using improved spectral libraries, which I'll show the data for towards the end of the presentation. And we're observing good quantitative accuracy with time-plex and also with this combinatorial time-plex plex DI. Okay, so next we wanted to do was apply this combinatorial approach to analyze single cells. And the reason being is that if you want to enable single cell analyses, there's many different analyses that you might want to do, such as profiling, understanding what phenotypes are heterogeneous in a cell, and seeing what proteins covary in abundance with that phenotype. And you can learn things about fundamental biological processes. To do that, you need ideally quantifying thousands, tens of thousands, hundreds of thousands of single cells. And so to do all that, you'll need to increase the throughput. And if you basically just rely on plex DI or time-plex, it's going to give you this multiplicative effect. It's going to be very useful. What would be even more useful is using this combined approach where you can really quadratically scale the throughput that you get by using both time-plex and plex DI. So that's what we're going to demonstrate today. So we prepared single cells by NPOP. This is a workflow that was developed by Andrew Leduc in Nikolai Slavov's lab. And we're going to analyze KIT 562 cells and U937 cells on a workhorse instrument, the Explorers 240, just as a proof of principle to see how good is the quality of data that we can generate from these single cells. At the same time, we analyzed 100x diluted bulk. So we have these cell cultures that are going, and we took off some bulk sample and we diluted it to 100 cells worth. And what we want to do now is assess the quality of this data, so we can use that bulk diluted standard, the ratio of protein abundances in the K cells to the U cells as our kind of guideline, and we can see what do the averaged out single cells, what did their quantitation look like relative to it. And the idea being is that they should agree, and we have strong correlation, 0.85, between these averaged out single cells and our bulk data suggesting we have good quantitative accuracy in our single cells. I'm highlighting two proteins, HMGA1, which is just a protein that's highly abundant, and FABP5, which is a protein that is about 5-fold more abundant in U937 cells than K562 cells. And I wanted to look at what the raw intensities were in these single cells for these proteins. And so what I'm showing on the bottom right is the total line chromatogram for these single cells. And then I am extracting signal at the MS1 level for these single cells. And what we see with the charged peptide, HMGA1, we have appreciable signal in all the single cells for this charged peptide. And there's no obvious differential bonds between them. But when we look at the charged peptide for FABP5, we have appreciable signal on all of them, and likewise we observe this differential abundance even in our raw data rather than just the averaged out single cells. We also did some dimensionality reduction, and what we found is that, as expected, we're getting really clean separation between cell types, and that we projected also the bulk samples into the same space as our single cells, and they are co-clustering as well. And so I think this shows a couple of different things. The quantitation of the single cells quality seems to be good, and also the methods of acquiring the data, one with time-plex alone, and also this combinatorial time-plex-plex DI are giving very similar results, which I think gives us more confidence in this technology as well. So to wrap this up, we have developed this orthogonal way of multiplexing, which we think can scale quite well, especially when paired with timeplex. And proof of principle, I think we've demonstrated the potential for an order of magnitude increase in throughput. And actually, if we scale this with the PSM tag that is a 9-plex, then effectively we would have a 27-plex. We can really rapidly accelerate the throughput of this and really reduce the costs and also the time required to generate such data sets. Okay, so this is more so about future avenues, which I'll keep relatively brief. The reason I was saying that we think that coverage can improve is, to me, something that is really intuitive. As we're using this joint modeling approach to model spectra of peptides, we have complete reliance on the library that we're given. And so if that library is even slightly inaccurate from what we're actually observing, we could end up with some lackluster results. And it's not because the data is bad, it's because our modeling is ill-informed. So to assess the impact of this, I looked at two data sets that I had, plex DI, which is 3-plex, and a 9-plex, which is a plex DI timeplex. And I searched this data with two different methods. One was a library that was really well tuned to the mass tag that we used. So you can consider this an optimized library. And the other one was a library that was reflecting label-free spectra, so it's a bad representation of what the spectra actually looked like. And if we searched it with the plex DIA, there's really only a 5% increase in the number of precursors identified with the optimized versus the unoptimized. And potentially this is because there's relatively low overlap, which Kevin was discussing earlier. So it's not a huge impact. But actually, as you go to higher and higher plex, that spectral library that you have becomes increasingly important. And so this is something that we think that we can further improve the coverage, just very simply having spectral libraries that more closely reflect what is empirically observed in that spectrum. And so lastly, the future directions of timeplex. The implementation that we have, we think works pretty well. We think that it can be improved in the future, and we're excited about future developments and what other people will think is good approaches to move this forward as well. One of these that I think is particularly exciting and should be accessible even today is work that was done by Rich McKibben in his lab. which uses a multi-segment capillary electrophoresis that temporarily encodes samples for capillary electrophoresis and for metabolomic analysis. And this should be usable even today for proteomic analysis, as far as I know. Another approach would be running isocratic separations using size exclusion chromatography, which would allow for a continuous gradient. Actually, effectively no gradient. It would just be the same solvent that's flowing through the same column, and you would sequentially just inject a new sample every X minutes. And potentially this would reduce overheads associated with time-plex. So a lot of future work that I think is really promising. So I'd like to thank in particular the team that made this possible, everybody at PTI, all of our funders, in particular Kevin for implementing this time-plex module into JMod, and Peyton for doing the single cell preps, and Nikolai and Harrison for guidance through this process, as well as I do want to say the preprint is coming along, should be pre-printed in the next couple days or so, next week or so, and we will have all the data available all the code available on GitHub. Data will be open access on our website and elsewhere. And if you have any questions, you can contact me at this email here. And thank you all for your attention.
Thank you, Jason. That was great. I see we already have a question, but as I make my way over there, I'd like to start off with one. Is that we saw that our PSM tag had really good scalability going from single to 5 to 9-plex. Do you think the not quite linear scalability is intrinsic in any particular aspect, or do you expect improved results with something like our PSM tag?
Yeah, I think anything that is going to improve the spectral quality is going to improve this analysis and this combinatorial effect as well. So I would expect it to definitely improve things. The trick, I think, is, especially for time-plex, is getting really accurate retention time so you can have that sample specificity. And that's going to help things along as well. Thanks.
Great talk. I wonder, maybe you've thought about this a little bit, but in the first talk, there was a bit of mention of how tags can substantially shift retention times. Have you thought of just maybe there's two totally kind of chemically different subsets of tags? One substantially delays retention times by, I don't know, 5 minutes or something, and then achieving a time-plex mode through something like that where you can just mix all the samples together, inject them, and then they just elute differently, and this allows you to do time-plexing.
So effectively have like different mass tags that you would pool together. Yeah, so that's a really good point. I think that would enable a multiplicative increase in throughput, but because the tags wouldn't be on the same peptide, it wouldn't be combinatorial. And so this would actually enable, this time-plex modality would enable multiplexing in time, because you could basically, you can do exactly what you're saying and go maybe from like a 3-plex for with M-track to maybe that you have a 9-plex PSM tag. You can combine them together, get a 12-plex. And then that wouldn't necessarily give you that combinatorial aspect, I think, if that answers your question.
So you need a different set of tags for each additional combinatorial.
Yeah, or you need to get, I think you would need to get the tags on to each peptide, right? You need to get a different tag onto each peptide to get that combinatorial effect, right?
Maybe not necessarily, or if you just had two totally, one tag that had 3 channels, and then a totally different tag that had 3 channels. And so now you combine your 6 samples together, and tag 1 was 5 minutes later than the other 3 channels.
Yeah. Oh, okay. So you're saying that they're like chemically
they're
the
totally
same?
chemically different, distinct tags.
Okay.
So you have like, you injected say M-track, you injected M-track labeled single cell in a sample with the PSM tag single cell labeled single cell. And say the PSM tag is more hydrophobic and generally elutes 2 minutes later per peptide. And now you have the PSM labeled and additional single cells for M-track, and you just totally treat them differently. I guess there would be other challenges.
I think that would be, would that be an additive effect? I think it would, right? Okay. We'll talk about it later. Thanks.
Hello. My question is a little bit more technical. You showed in a diagram that you're splitting the flow out and then going through 3 columns, and then you're either going into the mass spec via 3 sprays or basically backwards through your flow splitter again into a single spray. How are you going to implement that as you go up through your multiples? Because it looks like you're probably, or it sounds like since you're going 927, you're just going to take that flow splitter, multiply it by going through another set of flow splitters and another set of flow splitters. So I have 2 questions. One, how are you going to recombine them into a spray so that you're not just like blasting the front end of the mass spec with a bunch of liquid? And then also on the front end, you know, at what point do you think the LC pump is going to fail?
Yeah, great questions, great questions. No, yeah, we've had this exact same thought. And that's why we kind of went with 3, because it's the most usable. There actually potentially is a way to implement a 4-plex on this, but 3 is the most practical of this. Yeah, and like you're saying, potentially if we went to higher plex and we only have basically a cross, I think is what you're thinking about, where we have 3 inputs and 1 output. Yeah, if we wanted to scale this more, then you're right, we would need to have multiple of these crosses to basically join the flow. The way that we foresee this scaling in the very near future would be to have columns that hold very small volumes, so you can run very low flow rates without affecting your overheads. And then a result of that is that the cumulative flow that you get at the end of it would be hopefully not too bad. So that I think that in our mind is the way that we're going to go in the very near future. Thanks.
Related to that, if there's anything you wanted to expand on the challenges of implementing timeplex and how one might readily adopt it into their lab.
Thanks. Yeah, in terms of that, I think that is very similar. So the way that this can be implemented, you could join the flows. The thing that's really important when you join the flows is that the cross that you use has a very small void volume, as small as you can possibly get it. I think they can sell some crosses that are 50 micron bore size, and so it's a very small void volume that you would incur, because really you don't want to. You're separating all these chemicals on these columns that are really sophisticated. You don't want to basically create turbulent flow that's going to result in tailing peaks. And so just having connectors that are small void volumes so you don't disrupt the separations would be one implementation. There's also, you could use separate spraying approaches like we had demonstrated before. There are difficulties with that as well that make this practically a little bit harder. So I think the way forward probably will be doing joint flows.
I was wondering, how do you optimize the time delays?
Yeah, thank you. Thanks for the question. It's kind of interesting because it's very physically, the first assumption we make is that the columns are going to equally go back pressure, which for some columns that's definitely the case, for other columns it's not the case. And if you encode a difference in volume in those transfer lines, then theoretically if they hold the exact same back pressures, then you should be able to encode a very precise time offset. So like if one transfer line is 1 microliter and the other one's 200, and you flow, challenging my math, but anyway, you flow at a certain flow rate, then you're going to have an encoded time offset. And if the columns are relatively repeatable and robust, that back pressure shouldn't change, and so your time offsets shouldn't change too much. And that's basically what we've been observing too.
And a second question, so you mentioned library optimization at the end. What specifically about timeplex can you optimize for in a library?
Yeah, thanks for asking that. We can optimize, the most important thing is optimizing the retention times, so we can have a good expectation of when peptides are going to lead, so we can give them their sample specificity. And the other thing is that because we are searching this data using this joint modeling approach, having good expectations of how they're going to fragment is also really important. So it's a combination of both of those things. We can either, if we have an empirical library that has good retention times, basically you ran your sample, you searched it already, and it has retention times in your library that reflect what they actually should be, then you're pretty much set to go on the retention times. In cases where that isn't the case, we have built in a automated way of doing that fine tuning in silico. And so even if you don't have optimized retention times, you can still get pretty useful data. Thanks.
Related to the retention times and the optimization of timeplex, how small can you get in your delta retention time? And do you expect that it's a limitation of hardware or computational?
Yeah, thanks. That is a more so a limitation on the analysis side of things. So if you can get your retention times to be incredibly accurate down to within 10 seconds, there's really no reason why you can't offset samples by 20 seconds, for example. And so you could go to really, really short time offsets if you have really high precision and accuracy on your expected retention times. So it's really just a function of how robust the system is, so that they don't overlap in terms of their time, in reality, and then also how well you can model the data. So I think in the future, definitely down the line, I think we can go down to less than a minute.
Any more questions from the audience? Chat online. Nothing online. I'd like to remind our virtual participants to please do add questions to the chat. We are monitoring them. But if there are no more questions for Jason, let's give him another applause. Okay, so next we actually have our coffee break. We will begin our next talk at 4 o'clock. So please do enjoy some coffee and water. And I see there's some snacks back there too.
Do you want me to cut this shit for now? Yeah. I would say stop here. Yeah, I would say I
to introduce our next speaker, Mahlon Collins. He has a PhD from Pittsburgh University using proteomics to identify neurodegenerative disease biomarkers and molecular mechanisms. Then he did a postdoc at the University of Minnesota, where he investigated how individual genetic differences influence protein degradation in single cells. Now he is the Research Innovation Coordinator and AD Project Lead at PTI.
Thanks, Karina, for that introduction. Good afternoon, everyone. It's great to be able to be here and tell you about PTI's work on Alzheimer's disease that we've done over the last 9 months to look at novel protein dysfunctions. Now, I'm sure all of you are familiar with Alzheimer's disease, but I'll give a little bit of background just so we're all on the same page. You probably know that Alzheimer's disease is a relentlessly progressive, fatal neurodegenerative disorder. And perhaps you've even seen an MRI image like this, where if we look at a healthy control on the left and an Alzheimer's disease patient on the right, what is immediately evident is this striking atrophy of the tissue in the Alzheimer's disease patient. And that reflects widespread loss of neurons from throughout the brain in Alzheimer's disease. And unsurprisingly, that widespread neuronal loss is associated with a number of cognitive symptoms. Perhaps the most well-known is memory loss, but we also see things like emotional impairments and cognitive impairments in AD patients as well. So Alzheimer's disease affects at least 30 million people worldwide. That number is expected to at least double by the year 2050. We have no effective treatments for Alzheimer's disease. We have no way to prevent the disease. And so there's an urgent need for new discoveries in Alzheimer's disease to help us tackle this really immense problem that affects so many members of our society. And we think that PTI is especially well positioned to make those new discoveries in the space of Alzheimer's disease, because Alzheimer's and a lot of related neurodegenerative diseases are fundamentally disorders of protein dysfunctions. So pathologically, Alzheimer's disease is defined by two protein features. The first are extracellular aggregates of amyloid beta peptide. They assemble into these hairball-like clumps and accumulate in extracellular space. We call those amyloid plaques. You can see them in the brains of an Alzheimer's patient. The other feature that defines Alzheimer's disease pathologically are these intracellular aggregates, the microtubule-associated protein tau. So tau is normally bound to microtubules in Alzheimer's disease. It dissociates from those microtubules, forms these intracellular filamentous aggregates. And those occur widely within the neurons of AD patients. You can see them in dark brown in the top left there in an actual AD patient brain. So those features are actually how we diagnose AD pathologically. They define AD, and in some ways they've defined the way that we research AD as well. We focused extensively on amyloid beta and tau throughout the history since we've known about these proteins and their role in disease. But something I'd like to call your attention to is a theme that's emerged over the last few decades in AD research, which is that protein dysfunctions are actually much more widespread in Alzheimer's disease than simply amyloid beta and tau. So this is a non-exhaustive timeline of other proteins that we now know form pathological aggregates in Alzheimer's disease. And you can see that number has steadily increased over time. But I would also highlight about this list, in addition to the progress it shows that we made, is that most of these proteins were discovered in AD using targeted approaches. That is, we had a very specific hypothesis about this protein's role in disease, and we studied it individually in the context of Alzheimer's disease. And it's against that backdrop that I want to talk about what we hope to do at PTI to move forward our understanding of Alzheimer's disease. So I'll highlight two goals. The first is to comprehensively characterize protein dysfunctions in Alzheimer's disease. We know that even within a single Alzheimer's disease patient, there are numerous mechanisms that might give rise to protein dysfunctions. And again, a non-exhaustive list is highlighted here. And so to grapple with the complexity that AD poses at the individual level, at the population level, we need to be able to measure many patient samples. And because we can have protein dysfunctions arising from all these different mechanisms, they may touch many different proteins in the AD brain. And so we need to also be able to measure proteomes comprehensively in our studies of AD. The second goal that I would highlight for our AD research is to understand cell type selective vulnerability and disease progression. So these protein dysfunctions that I've been discussing, they don't occur in the same place in AD brain, and they occur with remarkable heterogeneity. And I think it's well illustrated by this schematic here, which shows the origin and spread of tau pathology here in A, and then amyloid beta pathology here in B, in the Alzheimer's disease brain. And you see that they originate in different places, they spread to different places in different ways. But even if we look at a very well-defined, anatomically precise and well-defined brain region, even within that single region, there is also a spectrum of vulnerability to these protein dysfunctions. There's a spectrum of vulnerability to whether neurons die or not. So in the schematic that I'm showing here, you see the 6 layers of the cerebral cortex arranged on the y-axis here. And then within those layers, you can see specific types of neurons here. And you can see from the color coding scale here that there's really this spectrum of vulnerability to death in Alzheimer's disease. And so to grapple with these complexities that AD brings with it, we also need to be able to measure proteomic changes within single cells and different cell types. And we also need to be able to make those measurements across multiple brain regions. So what we've done, what we've been doing at PTI is in support of these goals. And I'll talk about what we've done in regards to the first goal, comprehensively characterizing protein dysfunctions in Alzheimer's disease. And for this first part of the talk, I'll talk about some work we've done with bulk tissue samples. Before I get to the work itself, I want to highlight the people who have made it possible. First, the work is an ongoing collaboration with Brad Hyman's lab at Massachusetts General Hospital. A really productive and satisfying collaboration, both in terms of the resources we've been able to share with each other, as well as the conversations and the ideas that have come out of the collaboration. I want to highlight Greta Friedrich, our mass spectrometrist. She's done a great job with sample prep for these projects, and has also run the data on the mass spec and done a lot of great data analysis. Jason Derks has also helped out with sample preparation and has come up with some really creative analysis that has helped us interpret our data. And that latter thing can also be said for Maddy our computational scientist, who's also really helped us and analyze this data and come up with ways that we can interpret it that are biologically meaningful. We started with a cohort of 24 samples of AD and non-AD patients. We're profiling a region of the brain known as the dorsolateral prefrontal cortex. It's known to be involved in Alzheimer's disease. And our mass spec approach that we're using is the plex-CA approach that we've heard about throughout the day. Now I'd point out that we're using the M-track labeling approach here. We've heard a lot of exciting results related to PSM tag today, so you might be asking why don't you use PSM tag? We've been trying to build up our AD research efforts in parallel with the work that's happened on PSM tag. And so this is sort of in some ways our first generation of work that uses existing reagents, but in the future we're excited to increase the sample size and hopefully the number of proteins that we see by using the tags we've developed at PTI. So if we look at our results just at the differential protein abundance level, unsurprisingly we can see hundreds of proteins that are differentially abundant between AD and non-AD samples. And many of these reflect known changes that occur in Alzheimer's disease. And here our work has benefited from those who have come before us that have done large-scale proteomic profiling in AD brains, particularly the work of Junming Peng and Nikolai have helped our interpretation here. But we see things that we know change in Alzheimer's disease, and so we think we have a good data set here. ApoE is the largest known genetic risk factor for Alzheimer's disease. Complement C4a is an inflammation-related protein that's known to increase in Alzheimer's disease. And then conversely, we see decreases in cytoskeletal proteins, another common alteration in AD, and then also changes in signaling pathways. But we wanted to get a more interpretable view of the changes that we're seeing than something like this visualization provides. And in particular, we wanted to understand how proteins are changing within specific compartments of the cells in the AD brain. To illustrate how we did that, I'm just going to provide a simple conceptual example here. We can think about two conditions where our cells in condition A have fewer mitochondria than our cells in condition B. And if we do our proteomic profiling approach, On average, perhaps there is no fold change for all of the proteins in the cell. The average fold change here is 0. But if we were to look specifically at mitochondrial proteins and compare condition A to condition B, we would expect to see this leftward shift because there are fewer mitochondria and therefore fewer proteins there. So we, as a sanity check, applied this approach to our proteomic data, and we specifically focused on the cytoskeleton. So I said at the start of the talk that tau dissociates from microtubules in AD, so that leads to tau tangles forming, but it also leads to this collapse of the cytoskeletal structure inside the brains of AD patients. When we look at cytoskeletal proteins in the data that we collected, we see the expected large and significant shift in cytoskeletal proteins in AD versus control samples there. So since this reflects known AD biology, we use this as basically a scaling factor to look at other compartments within the cell. And we expand this analysis out to look throughout the cell, different organelles. And you probably can notice that one of the largest shifts that we see here is with the 20S proteasome core particle. Proteasome is the primary protein degradation machinery in eukaryotic cells. So we see a big shift in one component of it, but not in another. So to give you a sense of what that looks like, this is what the proteasome looks like, and it consists of two elements. The first is the 19S regulatory particle that binds proteins that are marked for degradation, unfolds them, and feeds them into the proteasome's 20S core particle, which actually degrades the proteins to short peptides, excuse me. So what we're seeing in our data is an imbalance where the levels of this aspect of the proteasome are decreased. These seem to be unchanged. And we thought that suggested to us that there are imbalances in the stoichiometry of this complex, which may affect the ability of the cells to carry out quality control protein degradation. And so indeed, if we looked at the correlations of subunits of the proteasome within non-AD cases at the level of both the 20S core particle as well as the 19S regulatory particle, we see the expected very strong correlations among subunits. This sort of correlation has been seen many times across many different systems. On the other hand, if we look at our AD samples, we see significant reductions in the correlation of proteins at the 20S core particle, as well as an even more significant reduction within the 19S regulatory particle. So what we're seeing here is a reduced stoichiometry of proteasome subunits, and that may reflect a reduced capacity of cells in AD patients to carry out essential protein degradation activities. And to see how that's affecting the proteins that we see that are of differential abundance in our samples, we specifically asked the question, are there proteins that are rising to increased abundance because the proteasome capacity of the cell is decreased? And to address that question, it's important to understand that proteins contain within them these regulatory sequences called degrons. Degrons can be bound by enzymes of the ubiquitin system. And when that happens, the protein becomes ubiquitinated. And then ubiquitinated proteins can be are marked for degradation and then bound and degraded by the proteasome. And so we took our list of proteins that we know are differentially abundant between our samples, and we asked, are they more likely to contain sequences that allow proteins to be targeted for degradation? And indeed, although the proteins that are differentially abundant have similar links, I'm not showing that data here, you can see that proteins that are increased in abundance are significantly more likely to contain a degradation signal than those that are of decreased abundance. We then asked, are there specific types of degradation signals that are in our proteins of increased abundance? On the x-axis here, I'm showing the ratio of proteins that are of increased versus decreased abundance. And then on the y-axis, I'm showing specific types of enzymes that recognize specific degradation signals. And you can see that there's a large and significant increase in this BAG6 complex. This result was intriguing to us because BAG6 is actually a quality control protein chaperone. It recognizes proteins that are mislocalized, proteins that rise to abundance perhaps because they can't be properly targeted for degradation. So we took our list of proteins that are recognized through this quality control mechanism, this BAG6 complex, and we asked what are the proteins that are of increased abundance there. And what stood out to us is this protein TMEM94. It's an ER-resident protein, but it also contains two of these BAG6 recognition motifs that allow it to be targeted for proteasomal degradation. And so because it contains these recognition motifs and because it's recognized by something that specifically targets mislocalized proteins, we think that this may be rising increased abundance through this decreased proteasome capacity in the cell. And indeed, we think that these recognition motifs that you see here are authentic bag-6 motifs. In a previous high-throughput assay that measures degron strength, you can see that as compared to the rest of the bag-6 N-terminus, they're significantly stronger in their ability to catalyze degradation, suggesting that they're true, authentic bag-6 motifs. And so we asked, in addition to the proteasome, this proteasomal defect that we've measured in 90 samples, might the targeting of Team M94 be affected by the stoichiometry of this complex as well? And indeed, if we look at the correlations of the members of this bag-6 complex, you can see that they, like the proteasome, are significantly reduced in our 80 samples relative to non-80 samples. So we think that this is a very specific protein dysfunction that's arising through quality control mechanisms that are likely impaired in Alzheimer's disease samples. And this is an area that we're really excited to follow up with more mechanistic work through our collaboration with Brad. So that's the work that we've done to characterize protein dysfunctions in AD. I want to talk about the second goal of our research, which is to understand cell type selective vulnerability and disease progression. And as I said at the start of the talk, for that we really have turned to single-cell proteomics. And we want to do single-cell proteomics on cells that we derive from human brain tissue. And before I get into our work to that end, I want to specifically highlight what Peyton Stewart has done to really move these projects forward. He's worked immensely hard and come up with a number of creative solutions that have really driven the research forward that have supported this effort. And so isolating single cells from human brain tissue poses significant challenges, as you can imagine. Although conceptually what we're going to do is very simple. We want to go from this to this. In practice, we perhaps undershoot it, as you can see here, or we can also overshoot it. And perhaps more significantly, the approaches that we have commonly used to dissociate cells, tissue to cells, are manual in nature. So there's likely issues of how much can we scale this, as well as how reproducible is it from person to person. You can also, an alternative approach is to use these protease enzymes. Those perhaps work well in the context of single-cell RNA-seq, but working at the level of proteins where these enzymes may have non-specific activities, it also poses challenges. So what Peyton has come up with, working together with folks from Nikolai's lab at NU, is an approach that is non-enzymatic and that's fully automated and standardized. We use this device that allows us to slice and grind tissue, that you can see here. And the result of this is that we get these morphologically identifiable neurons that have these nice processes that allow us to say, indeed, this is a really nice neuron that we've been able to isolate from this human brain tissue. Of course, it's one thing to see these nice-looking neurons underneath the microscope. It's another to be able to collect them. And so our approach for this is using the NPOP approach that was developed at Nikolai's lab at NU that we've heard about today. And we use an instrument called the Cell-in-One, developed by Sineon, to collect cells. And so it's been one of the things that's been really exciting with this project is to be able to not just see really nice neurons at the level of the microscope, but also to see them coming through the instrument as we're collecting them. So you can see we still have that nice neuronal morphology. We have projections that we can see that survive the process of being flowed through here. And we also can tell that these are neurons by staining for the nuclear marker, DAPI. And so with this, we can combine this fully standardized automated approach that we've come up with this NPOP-based approach to prep thousands of cells, thousands of neurons that we've collected here. And I would emphasize here too that the approach that we've taken is to focus on neurons. And we acknowledge that that approach has trade-offs. There are of course non-neuronal cell types in the brain as well, and those have interesting and probably important roles in diseases like Alzheimer's. But we also think that neurons are of clear biological interest in the context of AD. And that our ability to hone in on them makes this a really enticing area for this first single-cell proteomics project of AD and PTI. And so to this point, we've collected around 5,000 cells from across the stages of Alzheimer's disease. We get really nice distributions of cell size across the stages here, suggesting that we indeed are capturing neurons from throughout the stages of Alzheimer's disease. And more excitingly, we're getting what I think is really nice proteomic data from these cells. We get around 2,300 proteins per cell, And we get, excitingly, we get the big players, proteins that are of known relevance in AD. So tau, alpha-synuclein, this amyloid beta precursor protein, as well as TDP43. So we're getting lots of proteins. We're getting proteins that are of known interest in the context of AD as well. We've begun to start looking at data completeness, and we're also seeing encouraging results on that side of things. And then more recently, we've begun to look at how intact the cells that we are collecting are. And to do that, we're essentially looking for is protein leakage. So to what extent do the proteins from various compartments in the cells appear to be intact relative to a bulk sample that we might profile? And so you can see some analysis that Jason carried out for these single cells that we've collected. You can see on the x-axis is sort of the log2 fold change for the single cell data that we have versus the bulk. And you can see that overall, it depends on which protein you specifically look at, but we think that there's relatively little depletion from these samples. And I would say that these analyses are relatively preliminary. Data is literally rolling off the mass spec as I'm giving this talk, so we're still in the early stages here. But I think you can see one thing that is maybe not obvious from the data is that I think these distributions will tighten up as we look at more proteins and really build up these analyses. But preliminarily, we think we have relatively intact cells from this process that can be a really rich resource for AD research. So to wrap up here, I've highlighted a couple of goals for our AD research at PTI, and these are very much goals that we're in the midst of completing right now. We think we're getting the data. We still have more data to collect that can really build out and support the analyses that we want to do. But at the same time, I think the work that we've done so far is also shown that we can indeed find new protein dysfunctions that are occurring in Alzheimer's disease by generating high-throughput proteomic measurements that are comprehensive. So I'll specifically highlight the changes in the 20S and 19S regulatory particle subunits that we've seen in our bulk samples, as well as this change in this TMEM94 protein that is potentially mislocalized in AD. And then I think we also have a very clear path forward to understand cell-type selective vulnerability using single-cell proteomics that combines our approach for isolating morphologically identifiable neurons and the single-cell proteomic methods that are being developed at PTI. And of course, these goals are not independent of each other. We think that they support each other, and achieving both of these goals will be an important step forward to having a better understanding of Alzheimer's disease. Of course, with an interdisciplinary project like this, there's lots of people to thank. But first and foremost, we want to thank the patients and their families. They're the ones that make this research possible through their donations of samples. So we're immensely grateful for the opportunity to work with those. And then there's a few specific people to highlight here. At the PTI level, the mass spec team has helped us really get great data. Thibault and Lena have helped us learn to use the Cell-in-One. And Alex, I would highlight Alex Patelsky, who kind of kickstarted all of our AD research efforts at PTI. She's since moved on to an independent position. All the team at MGH always has great suggestions. They've been a rich resource for us. Andrew and Sina and you have really helped us with the sample prep side of things. We're appreciative of that. And also on the sample prep side of things, Josh Cantlin has helped us a lot as well. So we're appreciative of all that help, and I'm happy to take any questions.
All right, thanks, Mahlon. I'd like to open it up for some questions.
Thank you. It's really interesting work, especially with the Alzheimer's degradation pathway dysfunction that you discovered. And specifically, do you think you've potentially identified a mechanism that can be targeted with maybe some sort of drug?
Yeah, I think that there's a lot of interest not just in the context of Alzheimer's, but generally in how one can amplify these quality control pathways, right? So like alternative fasting was kind of a fad thing, and one of the hypothesized mechanisms there is that you're forcing your cells to do autophagy, which is sort of another route for protein degradation. So I think generally there is a lot of interest in increasing the activity of these clearance pathways, because we know that as the brain ages, there are multiple things that sort of accumulate more and more damage, and that leads to sort of an increasing loss of protein homeostasis that occurs over time. So I think that is sort of a fundamental mechanism of aging and also age-related diseases that I'm sure lots of folks are working on targeting therapeutically.
Yeah, sort of following up on the lack of protein homeostasis in the ADs. So if you look at other complexes not involved in degradation, do you also see them dysregulated? If you look at metabolic complexes or chaperonin systems, is it just generally like proteostasis is broken now all these complexes have dysregulated?
Yeah, we haven't done a deep systematic analysis of protein complex homeostasis within the disease, but my expectation would be that is the case, right? So these there's multiple pathways within the cell that are specifically dedicated to eliminating excess subunits of complexes that are produced. And so with the proteasome being compromised in these cells, with ubiquitin ligases also, the stoichiometry being off there, we expect that those sort of excess subunit accumulation things are happening. And all of these things are sort of accelerating the decline of protein homeostasis that's happening in disease, right? So so our expectation is certainly that those are altered as well.
Thank you. I'm wondering if, in the case of Alzheimer's, if you're going to look at other cell types in addition to neurons.
Yeah.
Certainly other brain cell types have been implicated. Yeah.
Yeah, absolutely. So one of the things I would say up front is that the approach that we've developed, we think, allows us to target any cell type that we want, right? So we can add fluorescent markers in, and so if you have a good marker for your astrocytes, your ligadendrocytes, things like that, we can go after those populations as well. The choice of neurons is somewhat based on the current throughput of the technologies that we have right now. So we think at current throughputs, a sort of because one can write up the tissue here, and I'm showing that we can get neurons from this, but we think that there's probably also intact astrocytes, ligadendrocytes, other cell types in there as well. The question is, can you sample enough of that to get enough of a certain cell type to be able to analyze it? And so we took data from the Allen Cell Brain Atlas, and we just looked at the abundance of cell types that they see, and we said, okay, if we sample at our current throughputs, what do we have the best chance of success with? And there the answer was very clearly neurons. But certainly going after cell types like astrocytes in a very targeted manner, we think is also something that can be of great value for the AD community.
My question is related. With the selective cell vulnerability, just looking at neurons, is the idea then that you'll go to some type of neuronal subtype, or what happens after you identify those pathologies that aggregate with the neurons?
You're asking, do we want to stratify the analysis further to specific neuronal cell types?
Yes.
Yeah, absolutely. And we think that the data can be our best guide in doing that. So we certainly can get out neuronal subtypes, but actually what we'd like to define is sort of where in the disease process are these neurons, right? So what would be really exciting from our perspective is to be able to kind of stage the neurons on sort of like a pseudo-time axis where we say, okay, this population of neurons is relatively early in disease, these are sort of at the midpoint, and these are about to die here. And then seeing how the proteome is changing across that axis, we think can be really informative, because we certainly have a good handle on what a dying cell looks like. I'm not sure that we have as great a handle on what a cell that's kind of early in that death process looks like, particularly within an actual human brain, as opposed to cells in a dish.
Any more questions from the audience? We do have one online. How applicable might the single cell data collection approach be to study other neurodegenerative diseases?
Very, very applicable, right? So, and our ability to selectively isolate neurons, I think, is really exciting in the context of other diseases like ALS. So in ALS, you have a very well-defined population, spinal motor neurons that are dying as part of that. And so you could imagine taking spinal cord tissue and selectively isolating motor neurons that way. We think it can be relevant for that. I would also add that these diseases, Alzheimer's, Parkinson's, and ALS, they actually share a lot of pathology. So like alpha-synuclein pathology is hallmark for Parkinson's, but it also occurs in a lot of AD patients as well. And so looking across the spectrum of these diseases, I think, can also be useful for understanding the proteomic drivers that sort of generally affect proteins that we know aggregate, and the ones that have very specific effects on something like tau versus alpha-synuclein. So we think the method is broadly applicable to understanding what's happening in neurons that are dying in these diseases.
Thank you. We do have one more, but I think you already answered it. It was how scalable is the single cell method to include other cell types. So we addressed that already. So in that case, let's thank Mahlon again. And finally, we have Keller to introduce our last speaker.
Hi everyone, thank you for being here. My name is Keller Gardner and I am a research associate on the immunology team at PTI. Today I have the honor of introducing Dr. Thibault Colombani, an astute scientist, insightful collaborator, and a very supportive mentor. Thibault is a senior scientist at PTI, where he leads the immunology team in leveraging proteomic technologies to understand how aging impacts the human immune system. After pursuing a PhD in immunology from the University of Nantes, he completed a 6-year postdoc in immunoengineering at Northeastern University. Today, Thibault will be discussing his work on single-cell proteomics as it applies to aging of the immune system in his presentation titled Single-Cell Proteomics, a Promising Tool to Broaden Our Understanding of Age-Induced Immune Alterations. Please join me in welcoming Dr. Thibault Colombani.
Thank you, Keller. So I hope that everyone still have some space for single-cell proteomics because my talk will be full of them. So I think that at this stage you all understand that at PTI we are really interested in understanding aging because aging influences a lot of biology from neurodegenerative diseases, as Mahlon just introduced to you. But in the immuno team, we're really interested in how aging impacts your immune system. And there is a lot of things impacted by aging, such as the organ evolution, so you will produce less immune cells, loss of proteostasis, a strong immune unbalance over time, immune cell senescence, a lot of your immune cells will die, as well as inflammation. And there is many consequences of all those aspects, but one of them is a global immunodeficiency. And I think that a concrete example of that we have all been through in the recent years is how our immune system answers to infections, either against SARS-CoV-2, which is the virus responsible of COVID-19, or influenza that you probably all have had during the winter because it's a seasonal flu. And if you look at this graph of mortality rate as a function of your age, you can see that the older you get, the higher your chance of dying from infection is increasing. And it's a simple fact that you're more vulnerable to infections when you get older. Fortunately, when you get older, usually you also get wiser. So when you look at the vaccine, the percentage of vaccinated people, when you're 65 years or plus, when it comes to flu vaccination, about 70% of the people are vaccinated as compared to 35% of the younger individual. However, vaccine fails to protect them efficiently, as when you're really young, the vaccination against flu protects you in 60% of the case. When you get older, it can drop as low as 10%. So not only you are more vulnerable to infection, but also when you get older, that vaccine protects you less efficiently. And so in our team, one of our projects is to try to understand how immune responses are impacted by aging and how vaccine responses are impacted by aging. And to do so, we started a collaboration with the CDC on flu vaccination to get access to a really incredible cohort of patients. And so far, we only screened 4 of them, 2 old and 2 young, that had been vaccinated against the flu. And we collected their blood before vaccination and up to 28 days after vaccinations, and isolated all the immune cells or PBMCs. And we decided to apply single-cell proteomics not only because we're a proteomic institute, but because there were a lot of reasoning behind that. First, the need to zoom in at a single-cell level, because not all immune processes are impacted evenly by aging, but also within each immune cells population, you have a strong heterogeneity on how they're impacted by aging. So we need to go down to a single-cell level to be able to get some answers. And also, as you are probably familiar with that, RNA abundances do not really translate well with protein abundances, but also by looking at protein, we're hoping to not being only able to look at protein expression, but also look at protein modification and also protein-to-protein interactions. And to build all this analysis, we decided to do a single-cell proteomic pipeline that rely on nanoproteomic sample preparation, or NPOP, developed by Andrew Leduc at Northeastern. Then to run all of those cells using prioritized single-cell proteomics, or PSCOPs, that has been developed by Mahlon at Northeastern University, and to conduct some biological analysis, and to give you some sense of numbers of what we have been able to do in this project. We are currently able at PTI to prepare about 2,000 cells per day for a total of 20,000 PBMCs that has been prepared in only 3 weeks. And all those cells has been acquired on an Express 480 in less than months. And also because we love challenges, most of you know LSL, for instance, what Mahlon has been talked about. But when it comes to immune cells, they're really, really tiny. So the amount of matures that we're working with is like significantly lower than most of the cells in the human body. So to go through all the steps, we first decided to do NPOP by combining TMT, which is an isobaric tag. One of the main reasons we use them was for the capacity to plex up to 32 samples together, and we hope that PSN tags will allow us to do the same thing in the future. And we use the 7-1 developed by Sayenians to do single-cell preparation, and we have optimized it thanks to the hard work of Lina Warner to be able to develop those 32-plex array, as you can see in the picture of the slide right now. So we were able to isolate cells individually in nanodroplet, to lysate, digest them, and also to tag them before doing single-cell proteomics. Out of those 20,000 PBMCs that we collected, some of them were cell debris, some of them were a clump of cells that just wanted to join the party, but most of them were really nice immune cells that we can analyze and identify. And so after some filtration to only keep the proper cell for our analysis, we got about 17,500 PBMCs to be run in our mass spectrometer. And then with the help of Sarah, we decided to perform PSCOP. And one of the questions that maybe you guys have is like, why PSCOP and how did we do it? Because there is many ways to analyze cell by proteomics and single cells by proteomics. And one of the main reasons is because PSCOP allows to analyze thousands of prioritized peptides across all the single cells. And also it maximizes instrument time. And for us it was super important because of the huge number of cells we're dealing with to be sure that we would have a really high sensitivity and a really high data completeness, as well as the best proteome coverage. And PSCOP allows us to have like an increased proteome coverage as compared to other techniques. And in a nutshell, PSCOP, as the name stands for, rely on building a purity list. And to do so, we first did the data-independent acquisition on the samples to build a purity list and to collect the retention time of each peptide to allow a software called Max Quantlife to know when to prioritize what peptides over another one. Then we did a couple of scoots using data-dependent acquisitions to be able to refine this list and to only keep within this list the peptides that we call efficiently prioritized. And then we finished by polishing it to only conserve the peptides that we were seeing consistently within all our single cells. And we took some decision making in building these priority lists because, as you can sense, a priority list means different tiers, and so how did we define them? And the first one is because I'm biased in immunology, the highest tiers contain all the proteins that belong to immune-related functions. And then we disperse the remaining peptides into 3 tiers based on their overall intensity. And also to maximize our chance to detect all those peptides, we increase the maximum injection time in our mass spectrometer for like these higher tiers containing all the immune genes to consistently detect them. And also we gave a little boost for like the lower abundant peptide to also be able to acquire them as often as possible. In total, we got about 1,100 proteins, with 20% of genes being on our higher tiers, that was the immune genes tier, 20% of them in the medium tier, 40% of them in the high tier, and 10% of them in the low tier. And how does it look like in practical? Max count life, every single time that we see a peptide, we'll decide if it will be sent for analysis in our mass spectrometer. And in red, you have the detected one, in green, you have the sent one, and you can see that for the lower tiers, only in 50% of the time, it will prioritize them. When it comes to the medium tier, it's 90% of the time, and when it comes to the higher tier, every single time you see a peptide, it will prioritize it and send it for like MS2 scaling. We did some quality control on our sample, and here you have the graph of the sum intensity of the peptide per single cells in blue, and in our negative control in red. Negative controls being just droplets that didn't receive any cells to look for the background of our analysis. And out of that, we filtered out about 30% of the cells that didn't match our quality standard, and we ended up having about 14,000 PBMCs that we analyzed for biological functions. It's led to having about 800 PBMCs per conditions and 3,000 PBMCs per patient. And as you can already see, for example, in the green side, we had to deal with a lot of like variability in sample qualities, which is the things that always happen with primary cells acquired from patients. In total, in those 48,000 cells that we collected, we had consistently about 2,800 peptides per immune cells, and about 950 proteins per immune cells, from which 240 of them were related to immune responses. And after imputation, we got up to 1,600 proteins per immune cells. And then, and this is really preliminary, we started to try to solve a puzzle with Megan that was what cell type was present into our sample. So, as compared to what Mahlon presented, where he was working with a pure population of neurons, PBMCs are actually like made of like 5 to 10 main cell types and a total of 25 to 35 different cell types. And we have no way to know what cell was dismissed in what droplets and received what tags. So to solve this huge and troubling puzzle, we decided to leverage a single-cell RNA-seq dataset published by Olin and All in Metro Communications that basically did the RNA sequencing of 1.3 million PBMCs for 120 patients. And we used this huge dataset to try to align our single-cell proteomic data and try to cluster our cells and being able to label them. To do so, we first selected the most highly variable gene on this RNA-seq dataset out of the top 3,000, and every single of one labeled here were linked to immune responses. We did the same from our single-cell protein dataset, and we got also like a couple of immune genes, but when we looked at the gene in common between both, we had about 240 genes that were highly variable and in common. And we used those genes to do clustering first of the RNA-seq data, and we're able to cluster cells in like 8 clusters, and to actually correctly label them with the main cell type presented to the PBMCs. And we did an asymmetric clustering of our single-cell proteomic data to try also to map them and to attribute them a cell type. Interestingly, we saw that overall the distribution of the cells in those different clusters was pretty similar, as the exception of the cluster 3 and 5. It can come from the fact that we work with samples at different vaccination stages, so it may have changed the cell population distribution. Also, 3 and 5 belong to the same lineage, the myelocyte, and maybe we are just not able to correctly cluster them with our current approach. So it's something that we're working on right now. But interestingly, we went one step further and we tried to confirm that the gene overexpressed in those clusters has some biological meaning. And we were first really happy to see that all our cytotoxic lymphocytes were clustering together, but also that they were overexpressing granzyme and porphyrines, that are the 2 most important molecules to trigger the functions to killing cells. Also, it was really interesting to see that CCL5 was the main driver of separation between NK cells and CD8 T cells, and CCL5 is a signature markers of effector cells. We also look at the monocyte cluster, and we're able to see that CD14, which is a monocyte marker, was overexpressed in this cell population, as well as lyse, which is an important enzyme to play a role in fighting bacteria and viruses. And finally, as another example, we found out that in our B cell cluster, we were able to follow the overexpression of CD74 and MCD1 that are respectively responsible to regulate B cell transcription and a lot of B cell function. So this gave us a lot of confidence in our approach of clustering the cells and being able to label them correctly in our single cell proteomic data. As a conclusion for this talk, we were able to collect and prepare and acquire about 17.5 thousand PBMCs in just a little bit over 2 months, combining NPOP and PSCOP, and to get about 1600 proteins per single cells. We successfully identified a couple of immune cell population, and we're still working on it to polish this work. And I hope that you will stay tuned for what is coming next, because right now we're working on trying to see how age influences immune cells, but also how the different patients respond to vaccination over time. And there is way more patient and single cell coming. We're currently processing 6 more patients from this cohort. And also we really hope to deeper the protein coverage of our single cell using the PSM tags that everyone presented to you today. So to conclude, I just would like to thank our founders, our collaborators, the CDC and Proteome Science, obviously everyone has PTIs that allow these projects to work. There is also more projects in the immuno team, so I hope that you guys will stay tuned for like the next results that we will share with everyone. And I thank you for your attention and we'll take any questions.
All right, thank you, Thibault. Any questions from the audience? I'll start with one. So this prioritized approach looked like it allowed we prioritized immuno-related genes, proteins specifically, and in one effort was to characterize those cell types, right, with the clustering as you demonstrated. How applicable do you think this approach will be for other types of cells?
That's an interesting question. I think that overall it's applicable to any cell type and any function. I think that the power of the PSCOP approach is to decide what question you want to answer by prioritizing those set of proteins, or those set of markers. So it can allow you to specifically identify some rare cell type by like prioritizing the detection of protein associated with them. It can also allow you to answer other questions, such as we could have taken this problem by another tail and decided to prioritize any genes related to aging. Or, for example, if you are interested in a specific pathway such as inflammation, you could prioritize any genes related to inflammations to be sure to see them in every single of your cells and to be able to draw conclusion out of like thousands of cells.
I might preempt a common question is, what about PTMs? Can we prioritize those? Did we look for those in these data?
So yes, we can definitely prioritize PTMs. I think that the challenge of prioritizing PTM at a single cell level is to be able to see them and to detect them. So, but in our data set, we have a couple of PTMs that we identify that we're trying to make sense of as of now.
Great, thanks. Any other questions from the audience?
How do you think about dealing with small cells that might have like really like little amounts of signal? How do you think about like dealing and handling with that versus like maybe saying it might be hard to discriminate whether it's like a failed cell or
Yes, that's a really good question. So one of the information that we can acquire from the cell one is definitely the size of the cell. So we can. And also we have a picture of every single of the cells, so that's something that we can analyze a posteriori, and also like to correlate the size of the cells to the signals, to be able to filter them in a better way. So definitely something is doable, but it's also like just challenging the full acquisition, right? Because there is so much peptides and proteins that we can detect per single cell, and the smaller they are, the more complicated it is to detect those peptides.
What do you think the best thing about like doing immunology through mass spec is? What is kind of, in your opinion, what does mass spec offer that you think benefits studying immunology through that the most?
That's a really good question, and that's probably why I joined PDI. I think that in the immunology field, there is so many unanswered questions when it comes to signaling pathways, or when it comes to like protein interactions. There is a lot of like redundant receptors that either use different signaling pathways or the same signaling pathways. Protein modification, how some protein modification could lead to like different immune responses, and so on and so forth. I think that there is so many unknown in immunology and so many answers that proteomics could offer.
Okay, if that's it for questions, let's thank Thibault. So next up we have our panel. So I'll invite Nikolai, Kevin, and Harrison to the stage. Let me get the microphone. So you've heard from everyone on our panel today. We have Harrison Specht and Nikolai Slavov, two of our co-founders, and then Kevin McDonnell, who was one of our first team members to join PTI. So we'd like to open up the questions again, both in person and online. I'll grab my computer. But I would like to open up, of course, with some questions, and I'd like to say that these questions can be anything technical that you've seen during the talks that maybe has come to you since our speakers have, maybe since our coffee break. But also, this is the second birthday of PTI. So if you have any questions you're curious about parallel squared, then please feel free to ask. But I'll get us started with kind of a general question that I'll ask each of you to answer. Is that since you've all been present for the entirety of PTI, can you share what you view as one of the greatest achievements, maybe personally or as an organization?
Sure, I can start. I think that since starting PTI, I think one of the greatest realizations has been bringing the team together. We're very fortunate to be able to bring together people with a very diverse set of expertise to work on a problem like the ones you heard about today. So I'm incredibly thankful for the team that has decided to join us on this journey.
I have so many things to choose from that I have to confess being challenged by that question in a way that I rarely feel challenged. So thank you for the challenge, Sarah. But I'm going to pick on two things. One is I'm really quite pleased as to the ability to detect increasing number of fragments for peptides labeled with PSM tag. I think it wasn't at all clear that this is going to be feasible to do by exploring relatively limited chemical space. And to me, that has been a very rewarding early observation that came on a year ago. And the other thing that I would choose to highlight is working with a team that is not afraid to tackle bold questions and to find that sometimes what you try to do is not possible. I think having this kind of attitude is very empowering, certainly something that I always enjoy myself, and being able to see this broadly shared within PTI makes it an environment that I tremendously enjoy working in.
Yeah, just to echo, I think the biggest thing for me is the team as well. I guess I haven't been part of setting up a lab before. This was a new experience, and seeing how it has grown, I guess, since I joined, and the caliber of people that joined and how projects can run in parallel and accelerate. And as you've seen here,
results in quite a short period of time. It's pretty exceptional to see. That's because of the algorithm execution, I guess.
Well, I you know, that was a bit of a reflection. So moving into the next, you know, several years, of course, and beyond, what are you most looking forward to at PTI?
You have the hot seat.
Yeah, no, I can't think of an answer. I mean, the easiest and self-interested answer is I'm really curious about the tags. Tag 1.0, tag 2.0. I think that, like Nikolai said, it's a really exciting discovery and gratifying to run a small assay on hundreds of compounds and find something that is working very well, and excited to expand on it. So technology-wise, that's my answer.
So what I would like to see happen is that we make proteomics much more scalable and extending far beyond the analysis of the abundance of protein products and more geared towards identifying other functional aspects of proteins, their modifications, localization within the cell, interaction with other proteins, and doing this not just for the analytical capability of detection and scalability, but really dovetailing this and driving those innovations with well-motivated biological problems. I think that being able to combine those two things together that are rarely coexisting in the same place is something that to me makes PTI very special. And I would like to see us doing this in a way that is compelling and helpful to others to adopt for their research. And that brings me to another part to this answer that I cannot resist adding, that beyond what I want to see happening at PTI, for me PTI is what's happening in the broader research community, not just what we do, but empowering others to start analyzing proteins directly so that we can increasingly replace indirect inferences with direct measurements, with data that are well positioned to start characterizing large populations of diverse patients so that we can detect and analyze comorbidities and have ultimately better understanding of disease mechanisms so that we can better predict outcomes.
Yeah, I guess from a computational point of view, I think we're generating now these large data sets, and the challenges that pose and all the new tools and maybe methods we need to develop, I think is exciting for me from a computational point of view. But then I guess we've heard about a lot of different technologies that we've been developing in parallel, to use the name, but now we get the opportunity to combine all these, so we can combine the different tags, we can combine the different analyses, apply them to these large single cell data sets, increase throughput, increase depth of coverage, and so combining all those together now is the next exciting chapter, I think.
Any questions from the audience? Andrew's got one, so I'll go.
I can shout. So at the outset of developing multiplex tags for DIA analysis, the question was always, how far can this be taken? How many single cells do you think you can run in parallel, at the same time, or kind of per unit time? There's a limit to how short you can make gradients. Has through your development of this first round of tags, do you feel like that an answer that has clarified a bit, or do you still feel like it's a little bit more work to kind of see how far you can push the limit?
I think that's a deep and insightful question that has aspects for all of us. But let me take a quick crack at that. So, to some extent, I think there is an implied aspect of it, a tacit aspect, how far we can push the multiplexing without seeing downsides in terms of coverage and quality of quantification, which is very much part of our ambition, what we try to do. Of course, if we answer the question how far we can push it, if we're willing to take compromises, the answer might be different. Now, in terms of how far we can increase multiplexing, there is the side of making the molecules. How many barcodes can we make by using new time-encoded multiplexing by using partially overlapping isotopic envelopes. I think my estimate for that is at least 100 without going to very complicated molecules, but as Jason showed today, we may not need to make that many molecules if we combine it with alternative orthogonal dimensions for multiplexing, such as time. Perhaps it can be more economic to make a 20-30 plex and combine that with a 4 or 5 time plex, and that would give us a really good perspective, very good capability to parallelize. And then, of course, there's a challenge of counting the associated increase in number of ions and data complexity dealing with that. I would actually leave Kevin to answer the data modeling aspect, but to add on the side of instrumentation, we have to worry about instruments having the capacity to count billions upon billions upon billions of ions. As I mentioned earlier this morning, the scale of the proteome is huge. And our friends at Bruker and Thermo Fisher come up with new and larger capacity, faster detectors every year. So I think at this point, capacity-wise, current commercial instruments can probably handle very well at least 30 plex for single cells, maybe 5-10 plex for bulk samples. But I see absolutely no physical limitations, practical limitations in the future to increase those capacities, and I am confident our friends are working on that.
Yeah, I guess to speak to the computational side of things, I showed, I think it was in my second or third slide, an ion map, and I guess as you multiplex, you're increasing the complexity of that ion map. Every single spectrum you take, you increase the number of peaks, and so as you can see with joint modeling, it inherently can model those if they're the exact same fragment. But if you have a low resolution mass spectrometer and you have a high peak next to now a lowly abundant peak, and are you able to model, I guess, that interference between them? And someone asked about the astro and high resolution earlier, and it's like, that can account for some of that, but as we exponentially or go to 100 plex, now we have 100 times more likely this interference happening, and so we don't know where the limit of that is. We haven't really seen that yet, but excited to see where it goes, but that may be a challenge for the future.
We certainly like challenges. It's very exciting to be on that forefront, pushing the limits and finding where they really are.
We have another question out here, unless Harrison can answer.
The only thing I'll add to what Kevin and Nikolai said is I think our theoretical expectations from the beginning, Andrew, were pretty similar to what they were 2 years ago, but I think we have a lot more clarity on exactly how we're going to test them. I think we've got some wonderful collaborators who are helping us design, and we are learning a lot about how to design exactly the tests, what we want to do to get that answer.
I have 2 questions. The first question is regarding to your studying Alzheimer's and looking into the disease mechanisms. How do you think, and this data is coming from brain tissue donated by patients, how do you think about testing it and your hypotheses in different disease models going forward? And then the second question, not related, is you're generating a lot of really rich data. How do you think about standardizing it for future machine learning models for diagnostics or things like that?
All right, I'm happy to take this one. I think the first one is about the challenge of testing mechanistic hypotheses from observational data. If we collect postmortem tissue from human patients, how do we test a specific mechanistic hypothesis? Perhaps the most readily available avenue for us is by using iPS-derived neurons, where we can do experiments. And we can also test it in animal models through our collaboration with Brad Hyman and other colleagues. Fundamentally, I think we have to acknowledge that when it comes to neurodegenerative diseases, there isn't a great model system because iPS neurons are not human brains. They're useful for testing things, but they're neurons in a dish. They're not brains. Mice are not humans, and one of the very pervasive growing awareness in the community of neurodegenerative researchers is that animal models, in some cases even primates, have major limitations in terms of recapitulating human disease. So we'll use the best methods available to the community, to anyone, primarily those two that they mentioned, but acknowledging that they do have limitations. And with regard to data standardization, we are very keenly aware of the importance of using strong benchmarks, establishing community standards. We recently led the first effort to develop best practices and recommendations for single-cell proteomics, working with other leaders in the community. And when we feel that there is a need to do that in the contexts of diseases or different data types, we would certainly initiate these community-driven initiatives to establish standards. But we are very mindful that we are using resources. We are fortunate to have access to these resources, and we should make the most of them, which includes making the data that we generate and collect available for others to analyze and interpret. That's a very important part of our mission.
Yeah, I just add to that in terms of a machine learning context. Like, nobody from a machine learning background wants to look at a load of raw spectra and say, okay, how can I train my model? So I think having that interpretable data and not just giving them the raw data, but actually something someone could use is super important, and which is something we have to do.
I think the final aspect we can add to that is the analyses specifically available to single-cell proteomic studies are well applicable to brain studies like Mahlon described on single neurons, single microglia. We're excited to apply those sort of analyses to the data we're able to collect with our collaborators.
Hello, I have a question. I heard you mention in future directions localization. I was wondering if you have an interest in adding imaging mass spectrometry or deep visual proteomics to your workflow in the future.
Yeah, these are interesting possibilities. I think in terms of localization, what I think more about is being able to either label with different mass tags proteins in different parts of the cell, because this works very well with our ecosystem. We can synergistically enable as part of developing other methods. To the extent that we can physically isolate different cellular compartments at high throughput with other methods, such as laser capture microdissection, that's certainly a possibility, even though those approaches have generally been difficult to adopt and scale in many people's hands. But mass spectrometry imaging, of course, is a wonderful method for analyzing lipids, metabolites. With proteins, that is more limited and harder potentially with peptides. But I think our foray in this direction and choice of method is going to depend on the specific biological problems that are motivating it, and what will be most effective and informative for the problem at hand.
We have a couple questions online, and I'll get back to the audience. One of them is a lot of clinical or translational labs use affinity-based proteomic techniques because of ease of use. How can PTI's techniques be made as accessible as possible for less technically experienced labs? And I think we talk a lot about community, so broadly, how can we get the community more involved? How do we make this accessible?
That is an excellent question. Would you like to answer, Harrison, or should I? All right. So let me take the first cut at that. So population proteomics, certainly in the context of plasma, is something that has been of great interest for decades and has received renewed attention and interest more recently, especially in the context of affinity reagents being developed for a larger number of proteins and being scaled to analyze samples from tens of thousands of patients. Mass spectrometry has the capacity to be much more specific in detecting exact amino acid use hundreds of thousands of samples, and this is exactly a place where our multiplexing technologies, our approaches to collecting data in parallel can make a huge difference. They can make the difference between something being technically possible but economically unaffordable to something that is able to not only increase data quality but also reduce price. And I think there was a very important element to that question, very astute, as to not only the feasibility and the price, but also the barrier to implementation. And when it comes to mass spectrometry, it's a very powerful, very sophisticated technique that requires expertise to understand it used correctly. And we can do two things in that regard. One is to educate the broader community. We have been very passionate for that for years, certainly in the context of single proteomics, more generally mass spectrometry. I think there's a lot to gain by explaining what are the capabilities and limitations and allowing more people to have access and use the technique. And the other one, of course, is having more state-of-the-art facilities that can help with this analysis, whether this is CROs or whether this is core facilities or other organizations. PTI is not a CRO or core facility, but we are certainly very happy to help to partner and to enable others to use our technology, to enable CROs and core facilities to do this analysis. And they think the combination of those different things, education plus facilities that are able to perform the analysis can make the technologies more accessible. And the final thing that I'll say in this lengthy answer, sorry, it turned out to be so lengthy, but it is a good and complex question, is that if we think of what happened with next generation sequencing and that being so broadly accessible and available, is that there was a sustained period of decades of major investments from federal and other sources that eventually resulted in very standardized and robust technology. And if one wants to get to a point of having robust and standardized technology, having this community engagement and long-term sustainable support is very helpful.
I'll add on to what Nikolai said, and that I hope that we're in the position to offer a lot of that. I think that not only are we trying to put out technology and open up platforms for collaboration, but given the position we are in as a nonprofit for forming collaborations, I think we're also in the position to well sustain them, to do the sort of education that Nikolai was talking about, not just writing papers to get an idea out there, but writing papers to get the idea out there, and then support the people who would adopt the idea, distribute it, use it. So I think that hopefully the model of this organization will prove to be effective towards those ends as well.
I may just add slightly, and I'm not sure if this was in the question at all, but from a computational point of view, I think with the tools we're developing here, from early on, we had this motivation, we wanted to make things open source, and I think it's super important that we continue to do that so people can see and understand, you know, what it is our models are doing and how they work and what is generating the data. And I was very lucky in my PhD from other groups had open source material that I could use and utilize and build upon. And so having that available to let people, you know, come into the field, see what actually an algorithm is, how it works, and then alongside that, the onus is on us now to also make it usable enough so that even you don't need the technical details to be able to run the source code. You need to be just something that's really accessible, really usable, and so the onus is on us now to provide that platform for people.
So at the beginning of the conference, you spoke about the space of billions of proteins to be analyzed, and some proportion of the billions of proteins have not yet been identified. And I'm wondering if you folks can kind of speak to how you're planning to identify new proteins. I see, you know, peptide fragments, or is this happening? Has it happened? Where do you see this happening in the future?
It has happened. This is a research project that actually took place in my lab here at Northeastern, and something that I'm very excited about. So it has to do with how do we know the sequences of the proteins. When we say that there is a human protein, we have database with the sequence. What is the origin of that database? The origin of that is a sequence of DNA usually, or potentially sometimes RNA, more rarely, that then by using a genetic code has been translated into a mono-expected sequence for the protein. And how do we know the genetic code? This is something that in the 1960s was worked out using in vitro experiments of synthetic RNAs translated by E. coli cell extracts. So this is not translation happening in vivo in mammalian cells. The E. coli translation apparatus, tRNAs, ribosomes are very different from ours. There are 10 times fewer transfer RNAs, and the ribosomes are different. So as to how are the proteins translated in the human body, it has been very difficult to do these measurements directly because identifying protein sequences is so difficult. Most of the time we don't de novo sequence proteins. We predict what the possible sequence is, and then we match that to existing spectra, and based on that we can tell, did we detect it, did we not detect it? And one of the reasons why this is so difficult is because our spectra have not had enough fragments to do the de novo sequence identification. To the extent that we are able to make tags and molecules that enhance fragmentation patterns, we are in a better position to do de novo sequence identification. So the data that we are generating, including the technologies that we are developing, are going to make direct protein sequencing much more powerful, and we'll start learning more and more about the full scope of sequences of human proteins. And we have additional clever ideas that we are very excited about that can further empower the ability to de novo sequence proteins without needing to have an existing hypothesis for what the sequence is.
I think I can close this out with one more question from online. The presentations described exciting methodological improvements in mass spec. Do you anticipate innovations or breakthroughs in MS instrumentation over the next 5 to 10 years? And I think we can preface that, being that we are not the instrument vendors. We can't anticipate, or we don't know what to expect from our vendors entirely. But as Nikolai already hinted to, capacity is one realm of possible road of improvement, especially as we increase this multiplexing. But I'll turn it over. I'm going to make you start, Harrison, and if you have anything else you want to expand on.
Thank you. Yeah, I mean, can this become a wish list?
Taking notes.
Yeah, thank you. Yeah, no, I think there's a lot of exciting possibilities. You saw our application of some of the, mass spec ancillary technologies like ion mobility in Maddy and Mark's presentation. I think technologies like those are very exciting in the sense that they offer more dimensions for analysis. We had to have a theme of the day and think about scaling proteomics. It's all these other dimensions for multiplexing that may be accessible. Jason well demonstrated multiplexing in the time domain. So I think that that's an exciting possibility for people putting together these LCMS systems.
Absolutely. I very much expect improvements in instrument hardware. So this is something that instrument vendors and very creative scientists, colleagues are constantly working on, challenging the capabilities. And I'm also very pleased to have seen a lot of enthusiasm in these colleagues and appreciation for the technologies that we're developing. They realize that this holds tremendous potential for increasing the capacity, the power of proteomics, which can open new applications, new markets. So this is something that is very synergistic and beneficial for companies as well as driving wider adoption, more scalable adoption of their instruments. Because if an instrument produces 10 times more data and has the same price tag, there are more customers for that instrument. And from that perspective, we work closely and discuss with physicists and engineers and corporate leaders at Brooker, at Thermo, what are some of the improvements that we think can really empower multiplexing and data applications. We can certainly benefit from the ability to handle more ions. As we multiplex more and more samples together, we need to be able to handle more ions. Gains in resolution are always welcome because they help us mitigate interference, which can become an increasing problem as we increase ion density. Certainly improvements in efficiency of transferring ions, converting them to detectors can give us very substantial gains in sensitivity. And that is important for single cell proteomics, for single organelle proteomics, for PTM analysis, being able to more efficiently capture fragments with different M over Z's. It works very synergistically with improving fragment generation so that we can ultimately detect more fragments and more confidently identify peptide sequences. So I think there is a large scope for improvement on the side of instrumentation, and many of those improvements are quite practical, feasible, and likely to happen within the next years and help us and help the rest of the community. Some of those improvements are going to take longer to be realized, but I'm certainly counting on collaborations with our friends who make the instrumentations that is required to achieve our mission, to make proteomics much more accessible and much more powerful so that we can drive discoveries and help understand diseases.
Yeah, I can give the short answer, more resolution, just agree with what's been said here, more capacity, and definitely that one of sampling more ions. I think particularly for this modeling approach, we talk about, you know, having that expectation is so important, but if you're sampling very few ions, suddenly you get into sampling statistics and you don't, it may be random or more variable what your actually fragmentation spectrum you see. So if we can get greater number of ions into the mass spectrometer, you have a better expectation of the fragmentation patterns and get more accurate identifications.
All right, let's thank our panel.
And now is the time to enjoy a reception, because remember, this is not just a symposium, it is really a celebration of our second birthday. PTI is turning 2! Thank you all very much for joining us today, and I look forward to more conversations and discussion during the reception session.